Welcome to Public Lecture 2015. It gives us a great pleasure to extend to you all a very warm welcome. Ladies and gentlemen, please enjoy this event from us. Lightning ready. Sound ready. Graphics ready. And this is Public Lecture 2015.
must be heading to the next place. So I have prepared a new strategy for getting new materials.
Assalamualaikum warahmatullahi wabarakatuh.
Good morning, ladies and gentlemen.
Greetings and salutation to the Honorable Dr. Andam Saifuddin as the head of Chemistry Department, Universitas Indonesia, and also for Dr. Ridla Bakri as the representative of Faculty of Mathematics and Science,
And the Honorable Professor Usman Sumo Parentangulan and Dr. Adli Aditya Parikesit as Supervisor of Metabolism Class 2015.
and also for our honorable guests for today's event, Mr. Yushin Kim and Mr. K. Jong from the Korea Advanced Institute for Science and Technology as our main speakers.
And last but not least,
Welcome to Public Lecture 2015, Biochemistry as Significant Knowledge in Our Living System.
First of all, let us praise to our Lord Allah. Because of His blessings, we are able to attend this event. And don't forget to praise to our Prophet Muhammad, who was guiding us into the right way of life.
Ladies and gentlemen, please allow us as masters of ceremony to introduce ourselves. My name is Mentari Dwi Astuti.
And I am Yusuf Zaid Hakim. We feel very honored for being a master of ceremony of this great event. We hope this event for today will be going smooth, well, and enlightening for all of us.
But before we begin today's event, we would like to welcome Ms. Fatia Fitri Musfiro to lead us sing the national anthem of Indonesia.
May we ask for all participants to stand up as we sing the Indonesian national anthem, Indonesia Raya, led by Ms. Fatia Fitri Musfiro. To all participants, please stand up.
You may be seated.
Thank you, Ms. Fatia Fitri Musfiro. Now we would like to review the following announcement for today's event. After this, we will have a greetings from our Project Officer of Public Lecture 2015, Ms. Siti Astari Rahmani.
Followed by greetings from Professor Usman Sumofren Tambunan as the Supervisor of Metabolism Class 2015.
Greetings from Dr. Endang Setudin, Head of the Chemistry Department, Universitas Indonesia.
Special performance by our friends of Metabolism Class 2015.
And we'll have a first lecture session by Dr. Eugene Kim.
And followed by another performance.
We'll have lunch and break time, followed by brief performance by our friends of Metabolism Class 2015.
And then we will have the second lecture session of by Mr. Ki Junjo, and then we will have after lecture entertainment.
And final tribute on Metabolism Class 2015.
And finally, the closing speech from Professor Usman Sumofren Tambunan.
Okay, before we start our events for today, you may be wondering what today's public lecture is all about.
This public lecture 2015 is an annual public lecture that is organized by Metabolism Class under the supervision of Professor Usman Sumofren Tambunan and Dr. Ali Aditya Parikesit. And this year we're also collaborating with Chemistry Department Class of 2012.
And this event is supported by RTC UIFM,
Suara Mahasiswa UI,
Era FM UNJ,
kabarkampus.com,
seputarkampus.com,
kampusupdate.com,
Radio Kampus ITB,
Sains UI,
seputar event.
and special thanks to Mrs. Etina Sution and Dr. Rila Bakri for the big support and the participation for this event. We also want to thank our alumni of Bioinformatics of Chemistry Department in Biochemistry Sub-Department.
Now may we call Ms. Siti Astari Rahmani as the Project Officer of Public Lecture 2015 to deliver her short remark. Ladies and gentlemen, please welcome Ms. Siti Astari Rahmani.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. First of all, let me introduce myself. My name is Siti Astari Rahmani, as a Faculty Officer of this public lecture. First of all, let us praise our Lord God, Allah Subhanahu wa Taala, because of His blessing we could able together here. Also, giving and salutation to Dr. Ridla Bakri, Professor Usman, Professor Kim Yushin, Professor Jong Hijun, Dr. Ari, also lecturers from Department of Chemistry. Good morning, everybody, ladies and gentlemen. Thank you so much for welcoming here. Okay, first of all, I want to say thank you to Professor Usman because of his kindness. We could organize this event. Also to Dr. Ari, thank you so much for your help. Also, Professor Kim Yushin and Professor thank you so much for giving us a chance to organize this event. Also to Dr. Ridla Bakri, thank you so much for your participation and support for us. And I also want to say thank you to my friends from Department Chemistry class 2012. Thank you so much for your hard work, for your big effort to organize this event. I hope through this public lecture, we could improve our knowledge about biochemistry research, and it could motivate us to give more contribution for our bureaucracy. Okay, I think that's all I want to say. We are very sorry if there's any inconvenience from this public lecture. Okay, thank you so much. Now, may we invite Professor Usman Sumostren Tambunan as the Supervisor of Metabolism Class 2015 to deliver his remark. Ladies and gentlemen, please welcome Professor Usman Sumostren Tambunan.
Morning. Dear Prof. Ki Jun Jao, Prof. Keo Chun Kim, my colleague, Vice Dean of Faculty of Mathematics and Science, University of Indonesia, Dr. Ridla Bakri, Head or Chair of Department of Chemistry, my dear distinguished guests. Let me introduce myself. I'm Usman Sumostren Tambunan, the Chair of Biochemistry Group of the Department of Chemistry, Faculty of Mathematics and Science, University of Indonesia. Our group has been working extensively on the utilization of bioinformatic tools for virology research. The object of our study are avian influenza, especially subtype H5N1 and H1N1, dengue, and human papillomavirus infection. As our study progresses, we eventually realized that the contribution of bioinformatics is a regional effort. It should somehow facilitate by an open forum that readily accessible to life scientists regional-wide. This forum will eventually disseminate the novel breakthrough in life science. I would like to thank you for coming to this event, especially to Indonesian participants. Originally, this event is always held by the Council of Students and is sustainable for some years until now. Through the years, we always provide internationally recognized speakers for this venue, mainly in the field of life science. Through the years, the advancement of Human Genome Project has provided insight on the functionality of human beings. In that respect, novel approach on coping the disease and medicine could be developed. Protein engineering has born in that area. One of our speakers today will deliver this topic. By providing techniques and instrumentation to optimize protein based on its amino acid sequences. Moreover, the drug delivery approach has been enabled by enabling the vehicle to go through various inlets in the cell. In this venue, we have secured to invited speakers from Korea Advanced Institute of Science and Technology, South Korea. They are Professor Ki Jun Jeong and Prof. Ye Jun Kim. Both of them are well-known experts in the life science. Thus, in this venue, Professor Ki Jun Jeong will give talk about protein engineering and Prof. Ye Jun Kim about transdermal drug delivery. We are delighted to be able to have such distinguished scholars join us, and are particularly happy because they are all able to stay for the whole conference this day. We are also providing soft skill enhancement in the venue. In this learning process of the student to work as an organizer, especially organizer of academic activity. Honesty and wisdom are two key words for having strong inclination of good soft skill. Once again, honesty and wisdom are two key words for having strong inclination of good soft skill. Ladies and gentlemen, kindly enjoy this public lecture. Stay with us and be happy. Thank you so much. Thank you.
Thank you, Professor Usman. Now, ladies and gentlemen, please welcome the Honorable Dr. Budiawan as the representative of Chemistry Department to deliver his welcoming speech, as well as opening this public lecture officially. Please welcome Mr. Budiawan.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. Especially our distinguished guest speaker, Professor Ye Jun Kim from Korean Advanced Institute of Science and Technology, and also Professor Dr. Ki Jun Cheong from Korea Advanced Institute of Science and Technology. Thank you very much for your coming in our session. And also, of course, in this special event also I would like to thank you, Professor Dr. Usman Sumo Friends. He has good organized this meeting. So we hope for this lecture from our guest speaker, we can have some benefit, especially for our department and also for our institution, Universitas of Indonesia. And the last, I think, will be benefit and big value for our nation. We hope this meeting also will be discussed about especially metabolisms and also especially in biochemistry. That's very important and play an important role in our life. I hope our distinguished guests will good delivery about his experience, his knowledge, and they can also share for us to discuss about it. Thank you very much. I would like to say at the end, I hope this event will good progress, and the end also we can hope some added value in this meeting. Thank you very much.
Thank you, Mr. Budiawan.
Ladies and gentlemen, before we start the lecture, let us welcome the amazing performance from Metabolism Class 2015. Please welcome Metabolism Class 2015.
So, the last minerals is in India. Wait a minute. Yeah, India, I have to go now.
Well, that was the performance from metabolism class 715. That was great, right?
Indeed. Now let us begin our first session lecture. We would like to welcome our moderator, Mr. Ahmad Hussein Akam.
But first, I would like to let you know who Mr. Ahmad Hussein Akaf is. Mr. Ahmad Hussein Akaf is currently enrolled in chemistry department in supervision of Professor Usman Sumafren Tamunan in Biochemistry sub-department. And he has also achieved honorable mention on National Chemistry Olympiad in Surabaya and has participated in International Natural Science Tournament in St. Petersburg, Russia last year.
He also got the best speaker
Yeah.
in the competition. Now, ladies and gentlemen, please welcome Mr. Ahmad Hussein Akam.
Okay, thank you very much for Miss and Mr. Master of Ceremony. Okay, today in this first session, we'll have a lecture with the topic of transdermal drug delivery. Okay, basically, what's transdermal drug delivery is? It's a way to deliver the drugs, but frankly, it's not as popular as oral delivery. So in this first session, we have a speaker from Korean Advanced Institute of Science and Technology, the distinguished speaker, Professor Kim Yoo-chun. And before we ask him to deliver his lecture, we will see his curriculum vitae first. This is Professor Kim Yoo-chun. He is an assistant professor in KAIST, and as you can see, his education in, I think, America, and the award and honor from Professor Kim, highest GPA award in USA University, and also he has many publications. So I'm very delightful to be a moderator of this distinguished speaker. So please welcome Professor Kim Yoo-chun.
Good morning, everyone. So I think this is the right way to answer good morning, right? This is Indonesian way, right? So my great honor is a chance to talk about my research topic in this one of the best schools in Indonesia. So yeah. So my name is Yoo-chun Kim. I heard that K-pop is pretty famous in Korea, in Indonesia, right? You know the Nick Yoo-chun, right? The same name, but different face start with that. So my research is about the biomedical research field. So pretty. I've changed a little bit about my topic today. So actually I sent out my topic about the transdermal drug delivery. So, but the scope is just too narrow. So I changed a little bit about the delivery and delivery and delivery. So all about the delivery. So I'm not sure how many people are interested about the delivery. My main research is about the delivery. So I'm mainly focusing on the delivery of vaccine, the delivery of drugs, to deliver some genes into the skin or your body or some your cells. So I will talk about overall about delivery tonight. Okay. So I think this is pretty. The audience is pretty broad, major. Some of you have a pretty deep knowledge about the biochemistry or biomedical research field, but some of you have no idea about what's going on in the biomedical research field. So I want to give a lecture as simple as possible for you to understand easily. Okay? So my research topic is about delivery. So I want to classify to 3 different types of delivery. So the first topic is about the actual my research field, transdermal delivery. So transdermal means, so one by one, trans means across, so dermal means skin. So we can deliver some drugs or any vaccine or any kind of molecule across the skin. So for example, when you go to the pharmacy, you have a lot of drug formulations. So usually you take the kind of oral drug, right? But you also have a lot of experience to get some ointment, right? Creams or some patch, right? That is all about the kind of transdermal delivery. So I want to introduce about transdermal drug delivery briefly, and also talk about my research field. So before I talk about the transdermal drug delivery, I am also interested about the overall drug delivery. So this is our humans. Usually we take the drugs by pills, right, or some tablets, or some ointments. Sometimes we get some drugs by injection using some syringe needle, right? Then the drugs are circulating in our body, and we can cure some disease or reduce the symptoms, right? However, most of the drugs, we take the drugs by oral or injections, their initial concentration of drug is too much high. That means it's too toxic for us, right? However, after that, our human body wants to clear out that drugs. So later on, the drug concentrations are going down. That's not good, right? That means if you have some problem in your body, in order to maintain the drug concentration in some level, you take the drugs every several hours, right? When you go to the doctor, and the doctor say, you should take this pill every 6 hours, every 10 hours, kind of thing, right? The reason is you should maintain the drug concentration in a certain level, right? So that's not good. So we want to achieve the controlled drug delivery or controlled drug release. So if we get this kind of drug release, kind of the blue color drug release profile, it's pretty good, right? You don't need to take additional drugs. You don't need to take a shot, right? So if you're just doing that way, it's pretty convenient. So in order to achieve this kind of things, many research is going on, and I think transdermal drug delivery can achieve this, right? Because if we apply the patch, drugs are sustained release into your body, so we can control the release of drugs pretty well, right? So that's the reason why transdermal drug delivery is really critical this time. So let's talk about the skin first, because we are talking about the skin right now, right? Transdermal means across the skin, right? So many people are pretty ignorant about the skin, right? I think most of the female students are pretty interested about your skin, right? You have a lot of apply the cosmetics and cream on your face, and also to do some like kind of make you beautiful, right? But most of the male student doesn't care, right? Many female male student doesn't apply any cosmetic in your face or your body, right? Same thing with me. So actually, what we are need to know about the skin in detail in order to understand about the transdermal protein. Okay? So this is a cartoon of the skin, and I think most of you have know about the actual anatomy of the skin. So we have a three very critical layer in your skin. The first layer is the straticoids in your outermost layer in your skin. Ironically, this is very thin. It's only 10 to 15 micrometer. It's almost kind of transparent film, like a vinyl film. It's very thin. However, this straticoium is very critical barrier. Right? So this can protect your body. Right? This is main barrier for drug delivery. So even though it's very thin, it can protect our body. So our kind of pathogenic molecules or any kind of molecule cannot attack your body easily. So if you drop some water droplet in your arms, they maintain about the droplet structure, right? That means the real water cannot penetrate across the skin, right? It's protected by your skin. So your skin is made by some lipid bilayer and some proteins, so they are pretty hydrophobic. Hydrophobic means your skin is not compatible with the water level. So your hydrophilic things cannot penetrate through the skin. That's one thing. As you can see the structure, even though the screen is too small, so it is hard to see that, but it looks like a multiple layer. This is a straticoium layer, even though it's very, it looks very dark, but it has a lot of multi-layer. That's the reason why the molecules cannot penetrate through the skin, through the straticoium. This is straticoium. So the next one is a bioepidermis. Just next to the straticoium. It's a little bit thicker than the straticoium, around 100 micrometer. And this is very critical, immunologically critical layer. So because there are a lot of immune cells. You know about the immunology, right? Kind of immune response. So they have some very critical immune cells, named Langerhans cells, inside of epidermis. This looks like this. And the last layer is dermis, around 2 millimeter, pretty thick layer. And this is also pretty very critical immune cell, named of dermal dendritic cell. So this epidermis and dermis, this layer is very critical immunological organs, right? So in my research or today topic, we need to talk about this by like this. So first, we are drug delivery person, so our main mission is to deliver across the skin, right? So first topic or first aim is how to overcome this straticoium layer efficiently, right? The second one is how to deliver some vaccine into this immunological layer, right? So in order to achieve these two, I will introduce some medical device that we developed in later, okay? So let's continue about the introductory part. So in transdermal delivery route, overall there are 4 routes available. First one is intercellular route. So just kind of using, if you apply your ointment, your creams, applying your surface of your skin, right? They're just permeate by passively, right? That is the intercellular route. In order to improve the permeations, we do some treatment of your skin. So kind of chemical treatments in your skin. I will talk about this also later. The second thing is the shunt route. Shunt route means you have a lot of hair follicle. Everybody has hair, so you have hair follicle. You also have some sweat ducts, right? So you already have some pathway inside of your skin. So using this kind of already formed pathway, we can deliver something to your skin. For example, if you apply some plus charge of drug here, and we apply some minus charge electricity here, then you can have kind of plus minus interactions. So we can pull down the plus charge drug through the shunt route. So the iontophoresis is the main device for delivery. And the third one is transcellular routes. That means we can make some instant formation of pore formation through the skin by kind of electronic shock or ultrasound. So electroporations or sonoporations, that kind of things, we apply some high voltage in your skin, and your skin is permeable for a very short time. After that, your skin are resealed. So in very short time, we can deliver something through the skin. So that is a transcellular route. The last one is most direct way. You can make some pathway inside of on the surface of your skin directly. That is microscale hole. So for example, my main research is microneedle. So you can kind of making some physical pathway inside of your skin. So you can deliver any kind of molecule through the skin. Okay? So today I talk about the delivery, especially about the transdermal drug delivery. I want to talk about this microscale hole using microneedle. Now later I will talk about this intercellular route using some chemical enhancer. Okay? So this is kind of why transdermal drug delivery is critical. So first, we can provide a constant low level in plasma for drugs. As I mentioned, that kind of control release, right? When you apply some ointment or patch, right? Drugs are controlling release into your body, so you can maintain the drug concentration. Right? That is pretty good advantage. Or we can avoid the first pass metabolism in the liver. That means if you take a drug by oral, right, most of the drugs go to your liver and it's decomposed. Right? So sometimes you lose the drug's activity. Right? That's not good. That's the reason why there's a lot of toxicity or side effect. So some drugs cannot be taken by your oral. Right? So we can use transdermal patch so we can overcome this problem. Avoid the chemical degradation, same one, and provide a predictable and extended duration of activity. So we can expect how much drugs can be delivered. Because if you don't need a drug anymore, you can just detach the patch. Then you can stop it. But if your oral drugs, after taking the oral drugs, you cannot control, right? You cannot wash out your body or your organs or your stomach. It's not easy, right? But in the patch, it's pretty easy. You just detach. Then you can control the drugs. The last one is suitable for patients who are unconscious or vomiting. Right? If your friends or your family are just unconscious because of some stroke or anything, they cannot take the drug by oral, right? The only way is injection. So maybe transdermal thing is also another option. And this is another piece. So this is a progress of transdermal drug markets right now. Even though compared to the oral drugs, it's pretty small yet. Only around 20 drugs are approved by the United States FDA. It's a pretty small market, but the trend is like this. They keep growing, numbers growing, and the market is also keep growing now. That means it's a very promising market, right? In future, more and more transdermal drugs will be available in the market, right? It's pretty promising. Okay, so let's talk about research in detail, okay? The first one is my main research field, microneedle. I'm not sure how many students heard about the microneedle. Raise your hands. Oh, around 5. Yeah, extremely low percentage of you guys know about the microneedle. Yeah, my research field, one of my research fields is microneedle. It's micro-sized needle, right? Very tiny needle. Why you need that one? So for transdermal drug delivery, we want to make some pore onto the surface of the skin. So we need some needle, but we can reduce the size of needle. Then we have no pains, right? It's good for you. So the microneedle was developed. Microneedle was first developed in 1998. It's almost 17 years ago, right? And right now, many research is going on about the microneedle. So I will talk about the microneedle first. So it's a very simple cartoon. Since 1998, 4 types of microneedle was developed. I think this is most direct and simple way. So name of poke and patch, just poke and patch. That means, as you can see that, you insert your microneedle into your skin, then take out. And there's some pathways of form, right? In your pore form in your skin, right? Poking, right? After that, we apply some drugs patch. Then the drugs can penetrate through the pathway. Very simple way, right? However, in this case, this is very easy to do it and a pretty simple way to do it. The problem is, we don't know how much drugs are actually penetrated or actually delivered. That's one disadvantage of this approach. So if your target drugs or vaccine is pretty expensive or is not available that much, then you cannot use this way. So the other 2 approaches available, coating approach and poking and release kind of dissolving type one. I want to explain one by one. The coating approach is in surface of a microneedle. You can coat your vaccine or drugs, right? Like this. Some drug coating vaccine or drugs. When you insert your skin, as you know that 70% of your body is composed of water, right? You have a lot of water in your body. Not blood, just water, right? So you have a lot of water content in your skin also. So you have a lot of body fluid. So we can make use of that one, okay? So after insertion, your COVID vaccines are dissolving off inside of your skin by your body fluid. Gone. Then we take out the needle. So COVID vaccine or drugs can be delivered into your skin. Right? The third one is poke and release. Actually, you can make your microneedle only made by drugs or vaccine. Right? So all microneedle itself composed of drug and some formulation. So this microneedle itself dissolving off inside of your skin, nothing remains. Just one patch, gone. Right? Very, very simple way to do it. Right? The last one is poke and flow. So kind of a miniaturized version of your hypodermic needle injection tool. Very tiny needle. in case of this, we can deliver some kind of a liquid form of vaccine or liquid form of drug. Right? The main problem is all these three using no infusion. Infusion means kind of a delivery of some liquid into your tissue, right? By some pressure, right? That means, I'm not sure how many people understand about this. When you go to doctor's office, your nurse injects something in your body, right? Your hip or arm, right? And he inserts the needle into your arm or hip. A little bit pain, but not much, right? But after injection something, right? Some liquids are coming into your tissue. Then you feel pain. Oh, something happened, right? That is infusion. Same one. When we insert something into your skin, you feel a little bit pain, but not much. But something are infused into your tissue, then you feel more pain. That means this microneedle kind of infusion of some liquid form of vaccine or drugs into your tissue, even though it's pretty nice technology, but they make some pain in your skin. So I think this two approach is very promising. And my research field is about this coating approach. So I will talk about this more in detail. Okay? Now I will show you some figure. This is like a microneedle figure about the poke and patch. So this microneedle is designed only for penetration, so making a pore. Right? It looks like a pyramid or some kind of some stick. Right? A lot of things, different type, silicones or metals or polymer. Right? Very different type of microneedles are developed. Right? Only for the penetration. Dissolving type one. Let's ask one more things. Kind of questions. If some material is pretty strong or strong enough when they are dry, but if they are dissolving or meet some water, they're gone. What's the material, do you think? Salt, yeah. Sugar, right? Rice, your favorite, right? Right. I also Korean, so we love rice. Actually, yeah, that kind of polysaccharide can be made for the microneedle, right? Even though we don't have a rice microneedle yet, we can make it, right? But actually, some researcher made the sugar for the microneedle. This one, F. This is made by the maltose, one kind of family of a sugar. They use maltose for microneedle like this, right? Some polymer, some kind of methylcellulose type of polymer. So all this kind of thing is pretty strong enough when they're dry. Right? After insert in your skin, your body fluid are weak and dissolving. All these are dissolved off inside your skin. Right? Pretty strong approach. But the problem is, as I mentioned, that all this microneedle is easily dissolved. That means before insertion in your skin, right? If your environment is too much humid and a lot of water environment around it, it's already dissolved off. That's one problem, right? So I think Indonesian people, this is not good. It's pretty humid culture, right? Anyway, so in order to overcome that one, this number G is developed. This is kind of a hybrid approach. You can see the metal. This is metal, right? And this is dissolving type. So this is different from that other one. So this is kind of a bullet type. Insert. And this red part are remaining in the skin. Take out. So this red part are remaining in your skin. Right? Kind of that was direct delivery. Anyway, this is dissolving type of metal. And it looks like kind of architecture, right? Kind of castle, right? Looks like that. All is about 100 micrometer size. You cannot see with your eyes, only in the microscope, right? All different type of structure, like this looks like a capsule, right? But this is around 200 micrometer, right? You cannot see by your eyes. And the main common thing is all are hollow type. They are vacant in the middle. That means we can deliver some kind of a liquid form into your body. Right? This is hollow type. This is my research field about the coating microneedle. It's very simple. Microneedle itself is made by some metal sheet. You have some stainless metal sheet. You draw something, cutting by laser, electropolishing. So you can make this kind of all type of microneedle easily. After that, we can coat the vaccine or drugs. The surface of the microneedle looks like this. This like a all type. This is coating one. After insertion, all gone, right? This is coating microneedle. So each microneedle has pros and cons, right? Advantage and some disadvantage. Okay? So solving type 1, this is only designed for an insertion. Right? It's easy to make it, just make it sharper. That's it. Problem is, drug loading and drug delivery, we can, as I mentioned that, we cannot know about how much drugs are delivered, so that's one big disadvantage. Coating or dissolving type is pretty easy, just insert, that's it. However, drug loading amount and drug delivery amount is pretty limited. So we cannot deliver a kind of much amount of drugs or vaccine into your body, right? That's one advantage, disadvantage. Hollow type, drug loading and delivery is pretty easy, right? Because insertion of your liquid form, there are no limitation of the drug loading or drug delivery, but it's pretty hard to make it. Making a hollow type, that's not easy. Okay, so we coat a lot of different type of molecule in the surface of microneedle. Okay, we can coat calcium. This is a small molecule, around 600 Dalton, very small, tiny molecule. Vitamin B, bovine serum albumin, around 5,000 Dalton. DNA, couple million Dalton. Some virus, couple hundred nanometer. Microsphere, insoluble BAS form. Microsphere, microparticle. All type of molecule can be easily coated on the surface of microneedle. Okay? So this is actual microneedle that was used in my research. And based on the scale bar, it's only 700 micrometer size. It's very tiny, right? I'm sorry that I didn't bring that one. So I want you to try and insert your skin, all the students, but sorry for that. When you compare an actual needle injection tool like this, hyperlocal needle, and this is a 5 microneedle patch. As you can see there, how small it is, right? You compare this one tiny needle and big hyperlocal needle. Okay, let's imagine. This is coming to your arm, this is coming to your arm. Which one you prefer? Right? This one, right? So that's the reason why microneedle is kind of attract a lot of attention right now, because this is good for patient compliance. Right? So, but not only for that kind of patient compliance, microneedle has a very strong advantage. Immunological advantage. This is conventional vaccine. You guys already have experience to take the vaccine, right? Influenza vaccine, hepatitis B, hepatitis C, malaria, all kind of vaccines you take, right? Problem is, you go to the hospital, your doctor inject your arms or your other body place, take this kind of vial, injection tool. Usually, these injection tools go to your muscle, kind of intramuscular injection, right? Unfortunately, your muscle doesn't have any immune cell, right? So you just deliver some vaccine into muscle. So that vaccine indirectly go to the some immune cell layer, so you can get some immune response. So this is not the direct way. It's not effective, right? So we want to deliver our vaccine. So we coat the microneedle here, and we can deliver the vaccine into epidermis and dermis layer. A lot of immune cell there, right? Deliver, boom, right? Many vaccines are going to your dermis and epidermis, and a lot of immune cell over there. So you can get improved immune response compared to intramuscular injection, right? This is the main advantage about the microneedle, right? So that's it? No, we have more advantage compared to conventional vaccination using intramuscular injection. First, I already mentioned about this patient compliance problem. Right? I think you also pretty young people, right? And you also have a lot of chance to take a shot, vaccine shot, right? But you're still scared about the injection, right? Frankly speaking, right? I also hating about the injection. It's not feel good, right? And also the main thing is the vaccine target. The vaccine target is infant, a small baby, right? I have two kids. One is 4 years old and another one is 1 year old. My second son take a vaccine, 15 different vaccines right now. Right? That means he needs a lot of shots. And my son always crying and scared and not happy, right? It's a big problem for patient compliance. Right? However, this microneedle is very tiny, less painful, almost painless, and your baby are not crying when you see the microneedle patch. This is just a small patch, right? So we can improve the patient compliance problem. Reuse of needle. Some country, I'm not sure Indonesia also doing it, I don't think so, but some country reuse of the needle injection tool. That means, ironically, in order to prevent some type of disease, some people are infected by other disease, right? It's very ironic things. But it's happened worldwide. So this kind of reuse of needle is a very significant problem right now. Microneedle is hard to reuse. We just want to use, throw away. We have no option to reuse this one. So we can also solve this problem. Cold chain. I think you guys also have experience to go to hospital when you have to take a shot in vaccine. Your nurse or your doctor take out the vaccine from the refrigerator, right? Because your vaccines are very sensitive to temperature. Right? Some vaccines lose their whole activity in the room temperature. That's the reason why we should put the vaccine into the refrigerator. But I'm not sure, some isolated region, there are no electricity, right? In Africa, you cannot imagine that there's electricity is available, right? So refrigeration is not easily available. So cold chain is another problem. But microneedle itself, this is dry form, so this thermal stability is pretty strong. Okay, I will show you about that data. I think this is the most critical advantage. I'm not sure this is also advantage in Indonesia. If you have to take a vaccine, you should go to the hospital, right? And your doctor, your nurse, or get inject something, right? That means always trained personnel, kind of doctor or nurse are necessary, right? But in this kind of microneedle patch, you can do by yourself. Just patch. You can order it. You can deliver it to your home or your office. You can open by yourself, apply by yourself. So you don't need to go to the hospital. You don't need to go to the doctor's office, right? So self-administration is possible, right? And vaccine shortage. I'm not sure about Indonesia also has been vaccine shortage from in Korea. 2009, the new swine H1N1 influenza was outbreak. We experienced about the vaccine shortage problem. We don't have enough vaccine for our nation. So vaccine shortage is another problem. If we use a microneedle, we can do sparing of dose. That means we can use small dosage of vaccine and we can have similar response. That means we can save the dosage. That's another good thing. Right? And volcanism. If the vaccine are like vials or syringe needle, it's huge space required for storing, right? But small tiny vaccine patch, it's pretty easy to handle, right? You can send it to mail, you can stock in your office, pretty easy to do it. So microsize is very have a lot of advantage. Okay, so I speak well of microneedle, okay? So why microneedle is critical and why this is necessary for human health. So I think influenza is another very critical disease in here, I guess, right? Am I right? Korea also have very significant problem with this. Last 2009, we have H1N1 problem. I think Indonesia also experienced this kind of influenza problem. So I strongly impressed about influenza. I'm still working on this influenza study. So we are targeting the influenza virus as our vaccine target. And as you know, the influenza is the RNA virus infectious disease. It's around kind of global particle like this. It's covered by some membrane protein, HA and NA. Is very critical. And it's still major health problem in Korea and worldwide. In 2009, new H1N1 outbreak, we have 200 people die and 9,000 people sick. It's not a big problem, but annually, WHO's report, 250,000 people die, or 3 or 5 million people sick every year. That's not good, right? More dangerous one is pandemic, a lot. I think avian flu is maybe a potential candidate for that one. I think Indonesia also has some severe problem with avian flu, right? As your students should be interested about avian flu. Because in some day, this avian flu will infect the human easily, I'm sure. In 2013, the most famous influenza professor in the name of Gaoka, who is a professor in the University of Wisconsin in the United States. Currently, this avian flu cannot infect the human easily. Only a human who touch or handle the avian species, they have some chance to be infected. But that person cannot infect his friend or his family. That's very good, right? That's the reason why avian flu is not that much dangerous this time. Only who touch the avian species, they are infected. It's good. But the worst thing is their survival rate, only 30%. Right? If you infect the avian flu, right? 7 or 6 or 7 out of 10 people die because of the avian flu. It's very pathogenic one. The good thing is, this avian flu is not infect human to human. That's good, right? But as I mentioned, Professor Kawaoka, who is a Wisconsin professor, he's an expert in the avian flu. And here are curious. Let's make mutation of avian flu, which can infect human to human. And he has success. So he, 2013 or 2012, he published his paper in Nature, and he found some species of avian flu which can infect human to human. He's a little bit crazy, right? If some terrorists obtain that species, there's a huge bioterror, right? So FBI and CIA are warning to him, don't expose your secret to anyone, right? Anyway, so someday these avian flus can infect human to human, and if that happens, big pandemic, it will be possible. So they can kill, I'm not sure. So in 1918, right, in human history, there's some Spanish flu was outbreak. And in that time, 50 to 60 million people died, right, because of that flu, right? But right now, the human population is doubling and doubling and doubling. So I think more people will die because of this pandemic. So it's very dangerous. So you should be cautious, okay? So that's the reason why the effective vaccine tool is necessary now, okay? So this is global pandemic, H1N1 outbreak like this, including Indonesia, also Korea, Japan, China, everywhere. Right? So influenza is very dangerous and easily spreading out all the right. So this is the influenza. So let's back to our research. We want to try to coat the vaccine on the surface of my griddle. Like this, we can coat the vaccine on the surface of my griddle like this. This is the same one. We staining the fluorescent dye in order to see the actual coating easily like this. And after insert into human skin, all gone. That means most of the vaccines are delivered into your skin easily. Okay? In order to conforming this like this, we cross sections of skin like this. This is insertion point, and you can see that most of the vaccines are locating inside of your skin. That means successfully delivered into your skin. Okay? How fast? Within 2 minutes, most of the vaccines delivered into your skin. Right? But the problem is, we found that vaccine is not that stable in the coating machine, like a coating stage. That's not good. Okay? After drying, and PBS means supposed to be buffer solution, and this is coating solution using some viscosity enhancer and surfactant and stabilizer. Viscosity enhancer is we use a dip coating, like a dip and take out, dip and take out, something like that. In order to coat something, we should have some viscosity enhancer, like a sticky one, right? That is a CMC. And surfactant means we want to coat the vaccine in a pretty uniform way. Right? That is a surfactant, stabilizer like this. The problem is, after coating, their activity is gone. That's not good. Right? Serious problem. We found that a lot of aggregation of this vaccine. That's a big problem, right? So we testing different type of stabilizer which can maintain the or retain the stability. And we found that trehalose, one of the family of the polysaccharide, they can retain the activity of the vaccine pretty well. So they also prevent aggregations. So they retain around 65% of activity. That's good. So we use this trehalose as our stabilizer. So we actually carry out an animal study. Right? Vaccine study always followed by the animal study. Because in order to check, okay, vaccine is working or not. Right? So we deliver vaccine. The IM means intramuscular injection, which is comparison, right? And microneedle, very tiny amount of 0.4 micrograms. So after deliver vaccine into the mouse, after 5 weeks, we do some challenge study. Challenge means we intentionally infect the mouse with live virus. Then we can confirm that the vaccine is working well or not. Right? If vaccine is working well, mouse can be protected, even though you are infected by a live virus. Right? That's the reason why we use the vaccine. Right? So this is very typical process about the vaccine research. So we do some challenge study like that. So I want to skip kind of too much detailed academic things. Right? I'm pretty sure you cannot understand fully about this. Even I was a very hard time to understand this. But I guess you know about the antigen and antibody response, right? You know? Antigen. So antigen means something, pathogenic things going to your body. Right? And your body recognizes, oh, something happened in your body, something attacked. And that antigen warning to your body, and your body prepares some weapons to fight against, right? That weapon is antibody, right? So antibodies form in your body. So we can check how much your antibody is formed by vaccine approach. And you can see this microneedle groups with stabilizer and ion groups. They show very similar antibody response. That means they successfully derive immune response. Okay? And also HIV titer is another functional antibody. Same way, microneedle and IM derive very strong immune response. This is actually monitoring about the whole health of your animal after live virus infection. Mouse study has some limitation, right? Mouse is different from human. That's one problem. Mouse are infected by influenza easily. It's good. The problem is there is no symptom. Humans have symptoms like some water level, right? And coughing, and raise your temperature is going up, right? A lot of you feel like some cold, like stuff like that. All kind of symptoms we have, but mouse doesn't have any symptom. They just lose their body weight. Gone. That's it. So it's one disadvantage. So anyway, we're measuring the body weight about the mouse. And we check the body weight, and you can see that microneedle with trehalose group and IM groups, their body weight retained. That means they're healthy. So body weight, 100%. They all survive. That means microneedle is pretty successfully delivered the vaccine and successfully derive immune response. Okay? This is another thing, kind of lung virus clearance. It's too much detail, but anyway, influenza is a respiratory infectious disease. That means they can attack your lung, right? So after infection, we check how remaining in your lung. Okay? So after infection, sorry for that, when you sacrifice a mouse, we take out the lung, we measure how much virus are remaining in your lung. That is lung virus titer. So if the lung virus is remaining much, that is not good, right? So control group, a lot of virus remaining is low scale. IM, intramuscular group, still a lot of virus are remaining still. But in microneedle groups, gone. Right? That means very powerful protection. All viruses are cleared out by your strong immune response, caused by microneedle. That's a good one. Okay. So this is one story that microneedle is very strong. Has this. As I mentioned that, sorry, there's something problem. I'm not sure, some computer compatibility problem. Anyway, so we testing about the kind of long-term storage. So even though microneedle is very powerful, if they lose their activity easily, that's not good, right? So we're just testing. We coat the vaccine on the surface of microneedle, and very bulky way. We just put it in your bench top. And after 1 week, and after 1 month, we inject the animal. And even 1 day, 1 week, 1 month after, with some stabilizer groups, their immune response is retained. Right? This is conformed by protection, also same way. Without any stabilizer, trials, 1 month, 1 week, when they lose their body weight very harsh. However, with Triados group, even 1 month after, their body weight is retained and their survival rate is almost good. That means microneedle technology is very good for retaining your vaccine stability also. Okay? I want to pass this one. We have not many time remaining, so this is kind of the message is we can spare dosage. Okay? I want to show just one data, like this. So I want to skip this part because of time limitation. But anyway, we compare the low dosage of microneedle here and high dosage of IM groups. If this and this is same response, that means we can save 3 times more vaccine, right? We're only using 0.3 micrograms, which is similar to 1 microgram IM, same immune response. That means we dose sparing 3 times. We can save. So one vaccine we can administer 3 person instead of 1 person. That's a good one, right? That is dose sparing, and we can see that a low dose to IM, a high dose to IM, low dose to microneedle, same in the same way in the protection. That's one thing. Okay. This is, I think you guys are interested about this because this is avian flu study by microneedle also, right? Same way, microneedle much better than IM in case of HIV titer, and protection 100% of microneedle and only 60% of IM. Okay? In case of avian flu, microneedle is working very well. Okay? How about the other vaccine? So I know this is a public lecture, and you guys are, I'm not sure how much people are heard about this kind of new concept. There are a lot of study working on the DNA vaccine. I'm not sure how many people heard about the DNA vaccine. Can you raise your hand? No one? Yeah, no one? Yeah, DNA vaccine is pretty, not that much new. It's firstly developed by 1990. It's almost 25 years old. The problem is there are no product yet, no commercial product yet. That means still a a lot of problems. Right? One critical problem is they fail in the clinical study. Right? That means DNA vaccine is not effective yet. So we are also curious about DNA vaccine and we are interested. DNA vaccine is different from just conventional vaccine. Conventional vaccine is just we deliver some virus or proteins into your body, and your body recognizes that protein or virus. Now something happens, something attacks your body, right? But DNA vaccine is different. DNA itself go to your body and go to the cell and express inside of cell, and some antigen proteins are made by DNA. So antigens are produced internally in your body, right? That's DNA vaccine. Why DNA vaccine is critical? DNA is easy to make, right? Compared to other virus vaccines. And also very safe, not pathogenic, right? So we also strongly interested about the DNA vaccine. So we want to confirm that DNA is expressed on the surface of skin. So we're testing about some person DNA first. And you can see that microneedle have successfully expressed some on the surface of DNA skin. And also do some animal study here compared to iron groups, some antibody, IgA titer, much better than IM groups. And survival rates, microneedle groups, IM groups, incomparable, right? Much better than the microneedle groups. Long virus titer also same way. And avian flu DNA vaccine study also carry out. Same way, microneedle is much better than IM. Just like this. Okay? So, a little bit changing gear, a little bit. So I talk about microneedle almost 1 hour. Yeah? I think you guys are a little bit boring. Someone are a little bit sleepy, right? So I want to change a little bit. I have 15 minutes remaining. I think my lecture will sign on 1 hour 15 minutes, right? I guess. So I want to use the remaining 15 minutes for other research topics. Okay? We'll talk about a lot of physical things. Some device, right? Medical device, microneedle, right? Something important. But sometimes micro device is not enough. So we want to do some other things. So we want to using a cell-penetrating peptide. As you know that peptides, you know, right? What is a peptide? This is my biochemistry class. What is a peptide? You guys know, right? Yeah, small chain protein, like kind of version of protein, composed of amino acids, right? So right now, peptide is very famous target in many research fields because they are biocompatible. This form of amino acid, right? Biodegradable. A lot of unique function. That is the reason why many people are more interested about the protein peptide. And we are also strongly interested for the peptide. So peptide can be used for skin delivery, drug delivery, protein drugs, gene delivery, and we are working on this. Today I will talk about the skin delivery and drug delivery. Okay. So peptide is like this, a lot of something like this. So I want to talk about the peptide for some chemical enhancer today. This is the first study that nobody did it before. So cell-penetrating peptide is around, it's just natural peptide, which is available in nature. And around 750 cell-penetrating peptide or antimicrobial peptide is available. Even you guys have some peptide in your skin. You don't know, right? You're a very valuable person, right? Because you have some peptide. Right? If you have some damage on your skin, you're not infected right after your damage. Why? A lot of immune response. Also, you have a lot of antimicrobial peptide or cell-penetrating peptide in your skin. So that peptide kill the virus, kill the microbial activity. Right? Anyway, we want to use that peptide, and the peptide has a function to make a pore inside of the virus membrane. Not virus, some microbial microbial species. So they have some function to make some pore. So we get some idea that the membrane of the microorganism, they are lipid bilayer, right? And our human skin, stratum corneum, that is also composed of lipid bilayer, multi-layer of lipid bilayer. So why don't you using this peptide for breakdown of the skin structure? Right? So we're testing around 20 different type of peptide enhancer. And we found that this one, red one, meganic peptide, is very powerful to improve skin enhancing technology, right? So what is meganic peptide? It's a very interesting thing, an interesting story. 1988, Dr. Gelsof, who is a famous guy in peptide research, he visited Africa, and he found a small pond. It was extremely contaminated. Right? You cannot drink. Color is all gone. A lot of virus, a lot of microorganisms. However, he found some frog from there. Frog are swimming, survive, looks happy. What's going on? So he's suspicious about this. Or why they're happy and why they can survive even that dirty pond, extremely contaminated pond. Right? And he gets a clue that he extract the skin, and he found this peptide, meganic peptide, from the skin of that frog. And because of this peptide, the frog's skin is protected against microorganisms. Right? Because the peptide kills all the microorganisms. So the frog will survive and they're happy. Right? So this meganic peptide was firstly developed in 1988. We want to use this meganic peptide if they have antimicrobial activity that can increase the porosity of the lipid bilayer. So we get this idea that, why don't you try using this in your skin, right? So this is make simple. Okay? So we want to try this. So only meganic peptide itself cannot penetrate the skin easily, right? So we combine with some chemical surfactant, N-rhodal sarcosine, which is an anion surfactant. You can see that the pink one is without, and the blue one is with meganic. This NLS chemical enhancer is itself is chemical enhancer, so they can improve the skin permeability, right? But when we combine this meganic peptide, we use it at the same time, they have huge synergistic effect. And we found that the red one is meganic peptide, which is tagged in fluorescent. And the PBS, no meganic peptide penetrates. In NLS, peptide itself go to the skin. But they are located inside of stratum corneum, which I already mentioned in the introductory part, which is the main skin barrier, right? So we're testing. Meganic peptide only locate inside of the stratum corneum. Okay? We also take a picture, a confocal picture. This is a very powerful tool because we can take an imaging of your skin according to the depth without any cutting or something, right? Optically, it is kind of just step like this. So depth is 5, 10, 15, up to 40, up to 100 micrometer, they can take an image like this. And he found that this chemical enhancer, they also helping the penetration of fluorescent molecule across the skin. But inside of, with the gaming peptide, they're locating only the threaded corneum, and they're helping more fluorescent molecule going into the skin, right? And the good thing is, this negative peptide is only locating in the threaded corneum. That's good, right? So we're suspicious. Oh, what happened? So we do analysis about many type of biochemical or biophysical things. This is a difference scattering calorimeter. We measure the lipid transition temperature. They're going down. This means that lipid structure is fluidized. That means your skin's lipid structure a little bit fluidized. That means more permeable. Right? Your striated corneum structure a little bit fluidized like this. And X-ray scattering, this is a crystalline cholesterol peak on your skin. When the cholesterol peak is gone or reduced, the lipid laminar structure is also collapsed. All your structure of striated corneum structures collapse by addition of NLS and NLS plus NA. And FTI structure also same way. They keep going up. That means the lipid structure is fluidized or disrupted. So we found that this self-parenting peptide or antimicrobial peptide, if they go to the skin, they can help disruption or fluidization of your structure. So, someone should. Excuse me. Okay. 5 minutes. Okay, so this ends with my pen. Still, this is my last one. Don't worry about this. And so, we found that negative peptides are locating only in the striated corneum structure, right? So they stay there, and according to like this, they're staying in the striated corneum. They're very strong plus charge, and I think this is the test we have all time. I want to show you my last one. This one. So I want to summarize all my study with this. So we talk about the microneedle, right? Microneedle. And DNA vaccine also. Right? As I mentioned that, this DNA vaccine should go into your skin cell, right? Not only skin cell. So we want to deliver some DNA vaccine across the skin first. After that, wait 3 minutes. I know. Don't worry. It's only 2 minutes. Okay, so and go to the skin first, then go to the skin cell. Right? We have 2 steps. But microneedle itself cannot carry out that. Microneedle help across the skin, but microneedle itself cannot help across the skin cell. Right? So we combine this microneedle with some electronic function, the microelectrode. So DNA vaccines are delivered to skin first, and by electroporation, they go into your skin cell. That's microneedle electroporation. It's kind of hybrid technology. The microneedle itself has a function as a delivery tool, but also microelectrode. So we testing about this for cell study, cell delivery. So we want to testing only the cell, and we apply some electric voltage like this, and we can deliver some calcium or magnesium albumin into your cell, and we found that, this is working well. Finally, we can also deliver some DNA, and also DNA screening to the cell. Okay? We're testing these kind of things. Okay. So we have a lot of different things, but I think we have enough time. I want to already use up enough time. So maybe in future I have another chance we talk about the totally different things. So let's finish my talk with my acknowledgement. So this is all my research funding sources. I have my research group is biomedical, micro and nano delivery lab. The name is pretty childish, right? But it can include everything. Biomedical, micro, microneedle, nano, nanoparticle, which is my research field, delivery, my research field is drug delivery. So BM&D is my laboratory. We have 18 students, 10 doctoral students, 7 master students, and 1 postdoc from India. He is an Indian postdoc. He's a brilliant researcher right now, and all is my students working with me. So I think I'm pretty on time. So this is my school landmark building. So I appreciate for your all audience, and thank you for listening.
Okay, thank you for your lecture, Professor. And because this next section we will have a discussion section. So, anyone want to ask? Yeah, Q&A section. Okay, please, Mr. in the back with the hand raised. Okay, please.
Okay. Okay, Assalamualaikum warahmatullahi wabarakatuh. Good morning, everybody. Good morning from KIF. Thank you for your good presentation. In your research, the one I get is your research is focused on drug delivery, right? Okay. You discuss about toxicity and concentration when we use mechanism of oral and injection in drug delivery. In here, we discussed about disadvantage from that mechanism, right? So, as we know, the microneedle we cannot see, so, just like microbe and another microorganism, right? And there are some methods to deliver the drug using microneedle by make another pore in our skin or using a pore that are already in our skin, right? So, what do you think? Because your presentation is limited by time. When we use the microneedle to deliver the drug, as we know, the vaccine is specific, right, to inhibit some microbial organism or virus. When we use it, as we know, our skin is have a defense mechanism to select or screening molecule or material that can in or cannot in and go out, like just like that. So when we use the microneedle, is it influence our system screening in our skin? As we know, there are a receptor and a carrier in our skin. So when we use it and put the vaccine, and the vaccine is specific, right? What I mean is,
we want to cure HIV patient, but if it influences the mechanism of defense in our skin, that the virus of Ebola or dengue can get to our skin easily by using that drug. We don't know. I don't know. Yeah. Can I speak Indonesian? Can you translate?
Okay, I think I can understand. So you want to talk about, so if you're using microneedle,
Yes.
another infection is possible.
Yeah, because in your presentation.
Pore inside the skin, so you worry about another infection. Right?
It influences the receptor and another mechanism in our skin, just like that. Because in your presentation using mouse, right? The condition and the environment is controlled, right? But like in here, we cannot see what is the what all the microorganism is happening.
Okay, Professor, basically he asked if the microneedle can interact with skin and can make another infection caused by microorganism or something.
Sure. That's a very nice question, even though it's a little bit long. Or very simple. Okay. So, make it short. Is it possible to infect another chance, right? Because we make the pore the surface of your skin, right? Fortunately, our skin has a very nice resealing system. If you have a problem in your skin, they want to reseal pore easily. Right? And there's one report at some researcher in the UK. He also suspicious about the same one. Microneedle insertion or make a pore in your skin. Is it possible to another microbial activity or microorganism invade through that pore? There's many people are also suspicious and worried. Okay? So he tested. He wants to insert microneedle in your skin. Artificially, on the surface of skin, which has a lot of pore, they're kind of scrubbing or kind of applying intentional microorganism, and you want to check any infection. Fortunately, no infection. That means there are a lot of reasons. One reason is our human skin is extremely elastic, kind of rubber, right? So, sorry, there's no dashboard or blackboard. So even though we make some pathway in the skin, there is elastic. Their actual diameter of your pathway is much less because of the elastic skin structure. So their actual pore size is much less than actual microneedle size. That's one reason. Because the pathway is too narrow, the bigger microorganism cannot invade. The second one is our human skin have very nice recovering activity. So our pores are resealed pretty fast. Even though big pore size is resealed, it takes some time. But microneedle pore size is very small, so they are resealed very fast. Because of that, microneedle is pretty safe. But in order to conforming about the safety, I recommend you to, if you want to use microneedle in your home, please using some alcohol cotton, scrubbing your administration site first, and you insert your skin and your microneedle. That's a pretty safe approach.
Okay, that's the answer from Dr. Kim. Any questions?
Don't make your fuss after answering.
Center. Yeah, please.
Thank you very much. Good morning, Prof. Kim. My name is Adam. I'm very
interested in research about the patch. Yeah. But I'm curious about the waste of the patch. The waste, the trash. If you say that if, people can really use the patch, right? And unfortunately in Indonesia, you can use it really everywhere, not in hospital. People can throw trash everywhere, and the patch and also the patch.
You are worried about the reuse of our microneedle, right?
I'm worried about the can the patch contaminate the environment? I mean, is it dangerous for environment? Because you say that the patch is contain like some DNA or something. I think maybe there will be some dangers if people just throw it everywhere, and it will make some environment change, like DNA mutation or something. Is there any prevention of that, or is there any special treatment for the waste of your patch?
Thank you for asking another question. So he asked me a question about after use of a microneedle, how to dispense. So because that is kind of a sharp waste, right? So most of the laboratory has a sharp waste container, right? So in my laboratory also, after we use of microneedle, we throw it into the sharp waste. But that's laboratory, right? So I fully understand that if you're using that one in your home, and if you throw away in your kitchen or whatever, your younger brother pick it up and swallow, that's a big problem. Right? I know that problem because I'm not an industrial person, so I didn't think about after kind of accidental events after use or commercialization, but I'm pretty sure that this is very tiny metal sheet, right? So every sharpness has the same problem, I guess. But we can throw into your trash can right away, it's okay. And also, if we want to really worry about that, we can collecting all the microneedle waste, because it's all about metal, right? So we can maybe reusable, not actual application, not only just material itself, reusable later. But that's very nice and key question, but frankly speaking, I didn't think about the actual, that kind of problem. Because I'm the academic person, I'm not a commercial company, so yeah, thank you for asking the question.
Okay, this discussion getting interesting. We already talked about the microneedle, the self-penetrating peptides, and the remaining function of microneedle, etc. And basically, what Kim has described the function of microneedle for vaccine. Anyone want to ask about the delivery of drugs using microneedles or something? Any question? In student, yeah.
Thank you. Please introduce myself. My name is Febrina Yodi from Faculty of Dentistry, Universitas Indonesia. All I want to ask you about that, everybody knows that sometimes in dentistry we also conduct anesthetics. So for the microneedle patch, is that possible that anesthetic material for oral mucous area can be used in the oral mucous area? Please.
Kind of anesthetic things.
Thank you very much.
She asked me about the kind of another application about the microneedle, discussion about the anesthetic application. So kind of lidocaine delivery, right? So very nice question. And also we carry out the lidocaine experiment. So we want to kind of local anesthesia, but actually not for dental use, for cosmetic use. So you guys, I'm not sure how many people care about your face. Yeah, many people care about your face. You have a lot of scar, thing, right? So if you treat about that one, you go to hospital and you maybe you need to do some kind of lidocaine creams or lidocaine injection, then you get rid of that spot or you can get rid of the scar, right? So we are collaborating with some small company in Korea, and we think about why don't you deliver that lidocaine using a microneedle? Right? So we successfully put the lidocaine to the surface of the microneedle, and we deliver it to the skin, and successfully can all the lidocaine delivered to skin. But unfortunately, there is no animal study yet, because animal is pretty hard to analyze. Animal feel pain or not, right? That's very challenging stuff. So the only way is maybe we insert something, anesthetic part, we insert or poke using some of your needles, like a big needle, and the animal makes some noise, right? If you feel pain, right? But there's not indirect, like first, it's not a direct way. So even though we already successfully delivered some lidocaine into skin successfully, but unfortunately we cannot confirm it yet about the actual animal study. So we have plans to do it. And I'm pretty sure that it also can be used for dental use the same way. Right now, all dental using anesthetic thing is by injection, right? Very painful, right? So we also, maybe you can use some micro patch in your dental report, I'm pretty sure.
Okay, we already talked about the other use of microneedles, the environmental case for the microneedles, and also, what is it? The first question is about care for your skin or another infection. Okay. Any questions? Mr. in the back. Yeah. Please.
Assalamualaikum warahmatullahi wabarakatuh. Allow me to introduce myself first. My name is Ari Aditya Purbali. I am a student in Chemistry Department, University of Indonesia. So, Professor, you discussed earlier today in your presentation that the types of micro needles, the polysaccharide type and the peptide type. Now, and you also discussed about when we want to inject the microneedles, we need to coat the vaccine. One of the methods is coating vaccine. Now, my question is this. Since we have two types, or probably other types, but you mainly discussed these two, the peptide types and the polysaccharide type, we want to coat these microneedles with the vaccine. So my question is, which one is more practical to coat the microneedles? Is it the polysaccharide type or the peptide type? Basically that's it, sir. Or is it depends on the vaccine?
Some misunderstanding. There's no peptide type.
Peptide micro needles?
There's no peptide microneedle. Sure.
Sorry. You mentioned about peptide. It's not the microneedles.
Peptide is a cell peptide, right? So there are four types of microneedle. Just make a pore. Second one is coating type, dissolving type, and hollow type. So you want to mention about the dissolving type and coating type.
Oh, okay. The dissolving type and my bad. The dissolving type or the coating. So which one is more easier to modify the vaccine?
Yeah.
Thank you.
With nice questions also. Yeah, I already mentioned that there is a coating microneedle is just a very powerful tool. Dissolving type is also not a powerful tool. The two thing is right now, I think mostly developed right now. Each has a pros and cons. Coating microneedle is very easy to make it. So just plain metal sheet. You can laser cutting, laser polishing. you can make it, right? And after that, insertion is also very complete. Because metal, sharp metal, is easy to penetrate any type of skin, right? Even the elastic skin, you can penetrate easily. So insertion and fabrication is very nice. But as one, the Boston, some Boston student, he asked me about the after you kind of hazardous effect, right? So that's one disadvantage. After injection, some sharp waves are remaining. That's one, I guess, one disadvantage. Dissolving type 1, very fancy, right? All vaccines are dissolving type. After insertion, all gone, very nice. The problem is, as I mentioned, that we cannot guarantee about the insertion. The sharpness should maintain, but this is dissolving type 1. So because of any humidity or any kind of environmental problem, it can be kind of dull, right? They cannot retain the sharpness. If that, we cannot guarantee about the insertion, complete insertion. So that's one problem of this. So each has a pro and con. So I cannot say that which one is better, which one is good. But I think both is good, but maybe we should keep developing about this.
That's it. Maybe it's related about how to process it and how to maintain it in production. Okay. The previous to us talk about self-replicating peptide. Maybe anyone want to ask about that because it's interesting also. Any question?
Hello, Professor. My name is Zana Adiati. I'm from Faculty of Pharmacy, University of Indonesia. And usually, transferable drug delivery systems won't get the systemic bioavailability in the human body as fast as intraparenteral or oral delivery, right? So I was curious if did you use any kind of loading dose or kind of penetration enhancer to get the right concentration in the body? And also, I would like to ask about, considering the stability of the vaccines, did you consider any possibility for other vaccines to use this kind of method?
The question is, other vaccines available?
Yeah.
Sure, I want to answer first. Right now, influenza is one of the vaccines which can be used in my cradle. So right now, polio, measles, hepatitis. Sorry, not yet. I didn't. It's not clear that first question. So the second question is, so any kind of virus, microbiome, any kind of vaccine is available for microbiome. So right now, I'm also working on some West Nile virus, measles, polio, hepatitis, all kind of vaccines available. So maybe in the future, more diversity will be achieved. I'm not sure. The first question is not clear to me.
So usually, transdermal won't get the bioavailability in systemic, as fast as parenteral or oral, right? So in this research of yours, did you use any kind of loading dose or penetration enhancer or any kind of method to get the right concentration, to get the minimum effective concentration in the body?
Frankly speaking, the cell-penetrating peptide study, I couldn't carry out the animal study. So I'm sorry that I cannot answer that. But many other research going on about the chemical enhancing agent, they can pretty successful to deliver adequate amount of drug into your body.
Thank you.
Thank you very much. Okay. We still have like 2 questions left.
Yeah,
Which one? Yeah.
you raise your hand
Every time.
7 times. So I should answer something. I should answer.
thank you very much for choosing me, finally. I'm really interested about this topic, transdermal drug chemistry. Now, street creation. Yeah, number one is these are the chemical drugs or vaccine. Traditional drugs like plant extract or everything used with this drug delivery system. Number two is how some allergy case, allergy case that could be happen when you transfer for especially corneal. How to avoid or handle it. How to avoid the allergy. Number three, how to calculate dose of drug or vaccine if you want to use microneedle for transdermal drug delivery.
So you mentioned about the
traditional drugs like plant extract or everything.
So right now, the microneedle is used for any type of drugs right now, kind of insulin or lidocaine or any kind of drugs can be used for microneedle right now. So there is no limitation for usage currently. So if you want to use any type of thing, you can use it. The second one is allergy problem. So allergy problem, I didn't actually think about that much in detail because someone has an allergy to metal, right? That's the problem. So maybe if that one, you need to use some dissolving time, right? But now, we are more concerned about the not allergy, but about the kind of some response from the human skin, right? Kind of redness or something like that. This is just typical. So sometimes that kind of some response in the skin and boosting the immune response sometimes. Right? So sometimes good, sometimes bad. So frankly speaking, allergy problem, especially because if you have metal allergy, I know some of you have some metal allergy, right? You cannot use that. Third one is dosage calculation. Okay, so that's also the final question for that. Dosage problem is very critical things, okay? Especially the vaccine. Your overdose of vaccine. We still have 5 minutes left. So the last person, she has 5 minutes more. I'm going to sing something more. Okay, maybe next. So, overdose is the most critical issue, especially vaccine, right? So if you take a lot of overdose vaccine, sometimes it's very pathogenic to your body. Very big side effect. So dosage control is very critical. In case of coating microneedle dosage, we have kind of calibration. So after coating vaccine, we know that we reported about how much dipping number and how the concentration of dosage in the coating machine. And we dissolving after coating on that needle, we dissolve in the buffer solution and we measuring about the actual amount. So we can calculate about the actual coating dosage like that one. Right? So then we can calculate how much dosage are delivered into your mouth or any kind of animal. That's the only way we can do it. It's kind of direct measurement of the coating from the coating machine. The coating.
Thank you. Yeah, I think the committee said that we have 5 minutes left. So, anyone want to ask? Yes, please.
Thank you so much for the opportunity and thank you so much for your presentation, Professor Kim. It was a very interesting presentation about transdermal drug delivery, and no wonder if I may say that your skin is amazingly flawless. I inquire you dry form. So the microneedle is in dry form, and how did you make it into dry form? That is one question.
Yeah.
Yeah.
First one by one.
Oh, okay.
I'm pretty old to memorize everything.
You don't
Okay, so how can you make dry form? It's just dry form, right? So, very easy. So it's coating machine. We can dip coating after take out, dry, dip, dry. So final version of coated microneedle is dry. All the formulation is dry on the surface of the microneedle. So it's no wonder that it's dry form. So because of that dry form, we can retain the stability of vaccine pretty well.
So maybe you spray before you put it in the machine. The material is sprayed direct maybe or.
No. Actually, this is deep coating one. So vaccine is in the coating solution. So we deep coating means dip and take out. Right.
I see.
Dip and take out.
Let it dry.
Repeat that one. Coating something on the surface. And after that, finally, it's just leave it. So it should be the dry.
So what about the hygiene issue? I mean, the conventional vaccine usually use the G1 to make the shelf life longer. What about this your vaccine? I mean, do you use some additional material to make?
Yeah, maybe we can study about or investigate that kind of thing also in the future. But currently we have no additional thing. The only thing is the trehalate, which is kind of stabilizer. If we are using that stabilizer, we can retain our activity up to 1 month. That's a good one. But maybe, yeah, that's good suggestion. We can add in another excipient to improving the shelf life of vaccine. This is really long run. Yeah. Thank you for a good suggestion.
Maybe one more question. I noticed that you mentioned about the safety of the DNA vaccine. What about the possibility of creating a new vaccine from once that DNA is inserted into the body? Okay. About the stability, I mean the safety of DNA vaccine. What about the possibility of creating new viruses of that DNA once it is inserted into our body?
What about
Yes, the possibility of that DNA is expressed into some kind of the life form, I mean the virus itself?
It's almost impossible. The DNA itself expresses only the protein. It's not converting the virus.
I see. So
yeah,
it's only part of the...
Yeah, antigen protein is expressed.
Oh,
so our human body are recognized only protein, not actual virus.
Not the entire genetic.
Only the protein itself. For example, influenza, HA protein is hemagglutinin protein. So we only make that protein.
Okay.
That's the reason why it's
Thank you so much for the explanation.
Okay. Professor, I think that's the last question because we use up our time. So let me summarize this first session. We talk about the customer drug delivery, basically mainly using microneedles as Professor Kim research, and also did review about the cell-penetrating peptides, and also the applications of the microneedles and its types, etc. It's very grateful to have you here and present your lecture. Thank you very much, and thank you for the audience to give high attention and good discussion for this first section. I think I will better be myself.
Thank you very much for.
And the second winner is for our moderator today.
Mr. Ahmad Hussein Akaf. And the So, Yusuf, what do you think about the lecture?
That's great, I think.
Yeah.
I think I have said it before. Yeah, that thing on your chest is just not the rose. Okay, I remember it. There is a stabilizer stone. May I borrow it? Okay, thank you very much. I have to go now.
Useless. Now, I have collected all of this material. I am the strongest man on that
Stop! You are not going anywhere!
2015. That was nice. That was great.
We should have a lot of performance to bring in our event.
Yeah, we have a lot of performance. Yeah. Do you want to know why?
Yes, of course. Could you explain to me why do we have so much entertainment
Well,
to bring in our event today?
Well, as you can see, today's public lecture is brought to you by the Metabolism Class 2015 and is under the supervision of Professor Usman Sumafundamudan and Dr. Arya Aditya, which has different kind of teaching methods.
Different kind.
Yeah, different kind, yeah.
How is it really different from the common or usual teaching method?
Well, when we have to give a presentation in front of the class, we have to give it in an art performance. Yeah, so it will be easier to understand.
Easier to understand.
Yeah,
Great.
that is really interesting method. Yeah. So, shall we start second session
Of
now?
course, and moderator for this session is Dr. Ali Aditya Parikasit. He is assistant lecturer of metabolism class. He got his bachelor and master degree in Department of Chemistry, Universitas Indonesia, and got his doctoral degree in Leipzig University in Germany. And currently is assisting Professor Usman in Bioinformatics Research Group. Now, ladies and gentlemen,
Please welcome Dr. Ali Aditya Parikasit.
So, good day everybody. How are you all? Are you ready for our next session now? Ready? Yeah. Okay, that's so. So, we are going to proceed to the next session, but let me introduce myself. I am Arli, so I'm very glad that now I'm here again, acting as a moderator for one of the sessions of this public lecture. I was did for the previous years. So, let us begin to read the curriculum detail of our next speaker. So our next speaker here is Professor Ki Jun Jong. So he is from the Department of Chemical Engineering of KAIS, and actually he finished all of his bachelor, master, and PhD in KAIS in Korea. And he has a lot of research experience. He started his postdoctoral fellow in the United States for some years, acting as a research associate, and then now acting as an associate professor in KAIS. And he has also a lot of publication in his disposal. So without further ado, so I will let me introduce now Professor Ki Jun Jong from KAIS.
Okay, thank you very much for your nice introduction. I prepared to reading the Aparut and Selamat Siang. Which one is good for this afternoon?
Selamat siang. Selamat siang.
Selamat siang. Okay, selamat siang. Okay, thank you. This is really great place. So this is first time to visit Jakarta in Indonesia. First, thanks for inviting us to this great place. So everything is great. The great scene and the great food. Everything is great. Also the performance is really great, very interesting. So also I like the K-pop and the first of course first generation. Also I like Yuna and Taeyeon. You know the Taeyeon's hometown and my hometown is the same. Okay. Also the great performance. The black masked man is a really great actor. I think so. Okay. I think that this is a very great festival. I think so. So I am really enjoying this festival, and I hope you also enjoy my talk. Okay. So my talk. Okay, the topic of my is the protein engineering. So I don't know whether, I think the major most of you made is about the chemistry or biochemistry, some in medical school. So I think you are familiar with this the protein. So today I'd like to give some general introduction about the protein and protein engineering, and also I'd like to show some successful example of protein engineering. So what's the protein? So protein is the major component in the cell. So it consists about the 50% of the dry cell weight. So as you know, the protein has, there are many different proteins. There are also the whole protein, they have different structure, different conformation, and also they have different function. So something important in the cell structure, so glycoprotein or collagen, so keratin, this here is also the protein, and also the protein, they have catalytic activity, so enzyme. So also some protein, they are involved in the transport. So transport protein or the regulation or some protective protein. So the antibody or the vaccine, so they are protective proteins. So there are very different proteins, they have very different structure. They are very essential in the cell. So why? So this is a simple metabolic pathway of a cell, so maybe the bacterial cell. So bacterial cells, for growth of the bacterial cell, they need to uptake the nutrient from the environment. So here's the glucose, one essential nutrient. How to import this nutrient from the environment? They use some transport system. So the transport system is also protein. So using this protein, they can uptake this nutrient from the environment, and then they are metabolized. First they are catabolized, and then they are also anabolized to synthesize some of the important building blocks. So nucleic acid and amino acid, so the essential building blocks can be synthesized. So for this synthesis, the nucleic acid and amino acid or the lipid, also they need some enzyme, very different enzyme, many enzymes. So all arrow, they indicate the biological reactions. All these biological reactions can be done by enzymes. So enzymes are essential to the industrial viability. Using this uptake and metabolic pathway, cell can grow. And also this tail structure. So tail structure is also consists of the protein. So using this tail, the plasma, also the bacterial cell can move. So these enzyme proteins, they are essential in the cell, the growth of the bacterial cell and animal living cell, and also the mobility. So they are very important. So they can be, so we can say the proteins are the workers in the cell factory. So without these proteins, this cell cannot grow, cannot produce, cannot synthesize anything. So they are really the heavy workers in the cell factory. So they are heavy workers, and also they are very smart workers. So from the nature, so we can get the very useful proteins. So and then they are used for the various field. So one example is the antibody. So today morning also the Professor Kim introduced this antibody. So antibody is very useful therapeutic proteins. So we can get this antibody from the nature, so human or the other animals, and then they can be used for the therapy and also the diagnostics and also the medical research or the biological research, and also the enzyme. So there are many enzymes which can degrade the biomass. So biomass means the hemicellulose, the cellulose. So there are many enzymes which can degrade the biomass. So by the degradation of this enzyme, so we can get some useful sugar. And then also the sugars can be converted to some useful things. So the chemicals, cosmetics, or biofuel, the bioplastics, all this conversion can be done by the protein enzymes. So in the last decades, many biological processes have been developed using these the proteins, enzymes, and then this is currently this is one big trend in the biotechnology. So like this, we can get many useful proteins from the natural source. But now, so we need to develop more the biological process for the new biological process. So also we need to use the new enzymes. So we need to produce a new product. So for the production of new product, we need to use the new enzymes. And also some biological process, they require some different conditions, so various conditions. Sometimes they require very extreme conditions, high temperature or low temperature or acidic condition. So we need to develop a new enzyme suitable for those extreme conditions. And also some process, so they require some long time reactions. So in the case, also we need some stable enzymes. So where we can get this enzyme, these proteins? First, we can get those enzyme proteins from the nature. But that's not enough. You can get those enzymes from the nature. You can get, but it takes a long time. It's not easy. So from the nature, so we cannot get all protein, the one we want to use. So in that case, so first we will screen the new enzyme from the nature, but it takes some long time, and also in most cases it's not easy. So in those cases, we can create new enzymes, new functional enzymes for the new purpose. So for the new biological process, for new conditions, we need to develop new enzymes. Okay? That's the protein engineering. So protein engineering is we design the protein structure toward the desired properties. So sometimes we need to change the substrate specificity, and also we change the binding affinity, and also we change the improve the catalytic activity. Also we can increase the thermal stability, expression maturation, and protein folding. So many different properties, many different protein functions can be changed. So that's the protein engineering. So here I prepared one great example of protein engineering. So some scientists, they found very interesting proteins from the jellyfish and then the sea coral. So from the jellyfish, this interesting protein, the green, the fluorescence of proteins were discovered. So from the jellyfish. So this protein can give a green fluorescence signal. And also from the coral, so scientists discovered this protein, the red fluorescence protein. So they can give this green color fluorescence and the red color fluorescence. They are very useful tools in the bioimaging. So using these colorful proteins, so they are useful for we can do the imaging, the bioimaging. So this is a mouse, and then the green fluorescence protein, and the red color fluorescence protein. Also this is the Drosophila embryo, maybe. So using the GFP and RFP, so we can analyze, we can understand what happened in the cells. Okay, they're very important tool in the current biotechnology. But just two color, it's not inconvenient. If we have more the colorful proteins, maybe we can do much more things. Okay? But just two color, so that's not inconvenient. So how we can get all the color protein, all the fluorescent protein, not green, not red. So we want to develop the other fluorescent protein. So where we can get all the fluorescent protein? To get the new fluorescent protein, should we go to the seed? So go to seed, and then maybe we can find the fluorescent protein. Yeah, you can find it. What? It takes a long time. Okay? Or sometimes you cannot find it. Okay? The time is problem. Okay? So scientists, they engineer these two proteins, green fluorescent protein and the red fluorescent protein, and they develop this rainbow color protein. So not in the seed. So in the laboratory, they engineer these two proteins, and then they developed this rainbow color protein. So name is very interesting. So honeydew, and then the banana, orange and tomato with the strawberry and cherry. So they develop very sweet fruits, not real seed. So using this colorful, so various colorful proteins, also we can do many things. So in the bioimaging or protein interaction study, they are really important, strong tools in the current biotechnology. Also, so using this colorful protein, also we can draw this great picture. So all other bacterial cells, they produce different color proteins, process protein. So this bacteria, they produce green color protein. So this bacteria produce the red color proteins. So okay, it's a very great scene. Or maybe you are familiar with this scene. So palm tree, and then the sun and some seaside. So very great pictures. So using this great process proteins, so we can do many things. So this is one great example of protein engineering. Actually, these three scientists, so they got the Nobel Prize in the 2008. So these two scientists, they discovered these two enzymes, the two protein, green plus protein and red plus protein. And this Professor Xian in the UC San Diego, he engineered these two protein and he developed this rainbow color process proteins. Okay. So like this, so by the protein engineering, we can create new enzymes which have the desired properties, some high stability or some long stability, and also the new specificity and the binding ability. So question, so how to engineer? So how to engineer the protein? Okay. So in most cases, the protein, the function, and the protein function and the protein properties, they are closely related to the protein structure and the protein conformation. So to change the protein function, protein properties, we need to change the protein conformation and protein structure. Okay? By changing the protein structure and the protein conformation, we can change the protein properties, protein function. So by changing the protein structure, some protein, they have new function, new properties. Or sometimes by changing the protein structure, they lose their original properties, their functions. So anyway, so by changing the protein structure, so to change the protein function and protein properties, we should change the protein structure and conformation. And next, how to change the protein structure? How to change the protein conformation? You know? How to the protein structure is determined. What's the basic unit of protein? The amino acid. So the protein structure, three-dimensional structure is determined by the amino acid sequence. And then amino acid is encoded by DNA. Okay, so DNA encodes the amino acid, and then amino acid they determine the protein structure. So to change the protein structure, the protein conformation, we need to change the amino acid. To change the amino acid, we should change the DNA. So by the changing the DNA sequence, that's the mutation. So by the changing the DNA sequence, so we can change the protein structure and conformation. And by changing the protein structure and conformation, we can change the protein function and protein properties. Okay, so to change to get the new protein, which have the desired properties, we should change the DNA sequence. We should give mutation. Okay. So how to the protein engineering? We give the mutation to the target the gene. So next question, how to give where to give mutation? So protein, the general protein has about 500 base or 1,000 base. So which one is the critical point? So we should change just the critical point and not the entire region. So in the protein, so the where to mutate. So this is the sequence of the target gene. So this is some long. So which one is the critical point? So we need to find the critical point for the mutation. But that's not easy. So this from this long DNA sequence, so we need to find the critical point. So to find the critical point for the mutation, so generally the two approaches are heavily employed. So one is the rational design, and then the other is the random design. So today, first I'd like to give some information about the rational design. So in some cases, so we have a structure information. So rational design is based on the protein structure information. So in some cases, we have this structure information so by some X-ray crystallography or some experiment. So we can get the protein structure information for the protein, and then more high resolution structure information, and also more the high resolution structure information. So in this case, so this gray color is a substrate. So from this structure analysis, so we can find what amino acid, what residue is responsible for the substrate binding. So maybe this amino acid or this amino acid, they are responsible for the protein binding or the protein catalytic activity. So to change the protein function and protein properties, we should change this the amino acid. So first we have the crystal protein structure information, and then based on the structure information, we can find the critical position of the mutation. Okay? So now there are big progress in the protein, the structure analysis, some of the modeling program. So using the modeling program, we can find some of the critical points. So for example, so these two positions are critical for the target binding or catalytic activity. So give the mutation to these 2 points, and then so we can develop new protein. So this is a rational design. So rational design is based on the protein structure information. If you have a very nice structure information, you can try this rational design approach for the protein engineering. But if you don't have structure information for the target protein, how can we do? So unfortunately, for much more proteins, we don't have structure information. So we cannot use this rational design approach. We don't have any structure information, so we cannot do this prediction of this modeling study. So we cannot find the critical position. Also, even though you have the structure information, some cases it's not easy to find the critical position. Okay? Because the current simulation and modeling system is not perfect. So it's not easy to find the critical position, even though we have the structure information. In also many cases, we don't have the structure information. So in those cases, we cannot use the rational design approaches. Those cases, so we can use random design. So we don't have any structure information. So even though we don't have the structure information, we can do the mutation. So this random mutation means the entire genes, they are randomly mutated. Or some specific regions, they are randomly mutated. Because we don't know where is the critical position. So first, so all entire genes, they are randomly mutated. So we can create, so here's the target gene. So we give the random mutation to the entire gene. So by this random mutation, so we can create a lot of the protein, the mutant. So they are all different mutants, they have different mutation. Okay? So from this protein mutants, so we need to isolate the best one. So by the screening of this protein mutant, we can isolate the new mutant which has the desired protein, desired function, desired properties. So in this This slide shows the overall scheme for the random design. So it's a target protein. So there we don't know the structure information. We don't have the structure information. So first give the random mutation. So we can, by giving the random mutation, we can create the big size library, mutant library in the bacterial cell. And then from this library, random library, so we need to do the screening to isolate the new mutant, which has the desired properties. Okay. So let's see the first step. The first step, random mutation. So to give a random mutation to the target gene. So there are several genetic tools for this random mutation. So error from PCR, the DNA shuffling, or STEC, or RASTIC, or several more genetic tools have been well established for the random mutation. Using those methods, generally the error from PCR or the DNA shuffling. So using the tools, genetic tools, we can give the random mutation to the target gene. So entire gene, they are randomly mutated. Okay? And then the general library size, the random library size, they are, after mutation, random mutation, they are transformed the E. coli bacterial cell, and then the bacterial cell library size is more than the 10 to the 7 or 10 to the 8 cells. 10 to the 7 or 10 to the 8, the different cells, they have a different mutation. So they're really big size, the library. So from the first we need to construct this big size library. And then next was the screening. Now we constructed a random mutation library. And then maybe the big size, 10 to the 7 or 10 to the 8. And then we need to find the best one, so which has the desired properties. So how to find the best screening? Okay? So let's see. So this is the big size library. So from this library, so we need to find the best one. It's easy. So look at this is agar plate. So now the E. coli cell, they grow on this agar plate. So how many cells can you see on this agar plate? How many cells? About 200 or 300 cells. Very small library. So from this library, 200 or 300, can you find the best one? Can you find? You can do it. This is very small library. Just 200 or 300. So how to find the best one? How to find it? Do you have any experience about the gene cloning? Please raise your hand. If you have any experience of the gene cloning. No? So about the 200 or 300 E. coli cells, so to find the best one, you should pick all clones by toothpick. So we generally use toothpick. Okay? So all the 200 or 300 cells, they are picked, and then they are inoculated some of the media, and then they are cultivated. And after cultivation, we can purify the protein, so different proteins, and then analyze the activity of proteins. Just 200 or 300, you can do. Maybe if you don't sleep, maybe you can do all this screening about 10 days. Maybe you can do. This is a bit small. But look at the real mutant library. How many? 10 to 7 or 10 to 8 cell. Can you do it? Can you find the best one from this big size library? If I ask this screening work to my graduate student, maybe they will be crazy. Angry, and they will say to me, bye. Okay, they cannot do. It's really difficult work. But we should construct this big size library, and then for the screening, we cannot use the traditional method. So to find the best one to screen the big size library, we should develop a very fast screening tools, very fast and systematic technology we should use. So beyond the two speeds, we should develop the high throughput screening. So using this high throughput screening, so my student will be happy. So like this. So for this purpose, to develop for the high speed screening, so one great tool is the FACS. The FACS is the fluorescence activated cell screening. So FACS is one powerful tool for the high throughput screening. So for this FACS, generally we use the flow cytometer, high speed flow cytometer. So in this flow cytometer, so there is this capillary line, and then here's the library, so mutant library, and then this flow cytometer has this radar system, some excitation, and also the detector systems. So here the mutant library, they are applied to this flow cytometer, and then the mutant library, if they have some fluorescence, so this the flow cytometer can detect this fluorescence. So if the mutant library have the different fluorescence, they can be excited by the radar, and then the emitted fluorescence can be detected by detector, and then one more things. So the high fluorescence cell, so this green color, the high fluorescence cells can be sorted, can be selected by some sorting systems. So from this the library, so they are analyzed by this radar and detector systems. Among this library, some high fluorescent cells, they can be sorted by these flow cytometer systems. So the screening speed of this instrument, the screening speed is very high, about 70,000 cells per second. Just 1 second, 70,000 cells can be screened. Okay, just 1 second, 70,000 cells. So the real library size 10 to 7 or 10 to 8 cells, they can be screened just in 1 hour. Just in 1 hour. Using this flow cytometer systems, the big size library can be screened very fast. So in this approach, randomization, so we can construct a random library by using some genetic tools. So we can construct this random library, so there's no problem. And then the high fluorescent cells can be screened by the flow cytometer FACS systems. So some positive clones, they can be screened out by this flow cytometer. But problem, so this is step. So we can construct this library, and then this instrument can screen the highly fluorescent cell. But it's a second step. How the cells can give the fluorescence. So the library was constructed, and then during the cultivation, they give the fluorescence. How they can give the fluorescence? So after cultivation, we added some fluorescent dye. So they are mixed with the cell library, and then among the library cells, some cells they are labeled with fluorescent dye, but some cells they don't be labeled with the fluorescent dye. So all the problems are selective label. You should do the selective labeling of fluorescence. So high activity clone, they should be labeled with a fluorescent dye. The low activity clones, they should be labeled with much less the fluorescent dye. So high active clone, they give a high fluorescence, and low activity clone, they give a low fluorescence, so they can be distinguished in this flow cytometer system, and then this flow cytometer system, they can isolate these high fluorescence cells. So high fluorescence cell is the high active clone, so they have the target, the mutant of proteins. So we should do this selective labeling of fluorescence. This is the most important step in the random mutation. Okay? The success of the random mutation depends on this selective labeling. So this library construction is not difficult, and also the FACS screening can be done by the high-speed flow cytometer systems. But we should develop a very smart system for this selective labeling of fluorescence. If develop a very smart system, so you can succeed in the protein engineering. So my research is also focused on these systems for selective labeling of methane. So today, I'd like to introduce three strategies. So strategy one is direct interaction of protein with the process ligand, and the strategy two is direct labeling of product with the process dye. Third strategy is FACS screening with the genetic circuit. So today, now, I'd like to introduce these three strategies for selective labeling, and also I'd like to show some successful results of protein engineering. So first strategy is the direct interaction of protein with the process ligand. So here's from the plasmid DNA, so target genes, they are transcribed and translated, and then proteins are synthesized, and then we add this the process dye conjugated ligand. So the ligand is a target molecule, and then this ligand is conjugated to the process dye, and they add to this bacterial cell. And if there is strong interaction between the protein and then the ligand, so this protein can capture this ligand. Okay? So by capturing this ligand, process dye conjugated ligand, now this cell they can give a high process. If there is a strong, if there is a very weak interaction between the protein and the ligand, so they can give very low the process intensity. Okay? So the process intensity of the cells are highly correlated with the protein ligand interaction. So strong interaction between the protein and then the ligand, they can give a high process intensity. But the protein and the ligand, they have some very weak interaction, so they can give a much lower process intensity. So according to the interaction between the protein and the ligand, the process intensity can be changed. Okay? So this different interaction, different process intensity can be analyzed by the flow cytometer, and then so we can isolate the best one. The best one is a high affinity to the target molecules. So one best model for this strategy is the antibody. Okay? So antibody can bind to the antigen. Okay? So here's a near the target antigen. So the antigen is the conjugate the process dye, and then if this antibody has a high affinity, if the antibody has a high affinity to this the target antigen, they can capture this target antigen, and then they can give a high fluorescence. But if the antibody cannot bind to this antigen, so the process dye conjugated antigen cannot be captured, and those cells cannot give high fluorescence. So they can be distinguished by flow cytometer. So generally, in this study, generally the antigens are some big molecules, generally virus molecule or antigen molecule is a big molecule. So big molecules is this antigen molecule is too big to be entered to the host cell. So generally we use this cell surface display. So antibody is a display of the cell surface, and then they can bind to the target antigen. And then if there is a strong interaction between the antigen and antibody, they can give a high fluorescence. Okay? They can be analyzed by the flow cytometer, and then we can isolate the high affinity, the antibody clones. Okay? So let me give introduce about the antibody. Today morning, Professor Kim also gave some introduction about the antibody. So antibody, we have 5 different antibodies, IgA, IgG, IgE, IgG, IgM. So most popular antibody is the IgG, about 75% of serum immunoglobulin. So it consists of 2 heavy chain and 2 light chain. Also, it has a very specific binding to the target antigen. So this can protect our body against the pathogens. Okay? So also, they are very important pharmaceutical proteins. So in the last year, maybe 2014, so the top that the best-selling blockbuster. So most of the antibodies, they are ranked in the top 10, the best-selling blockbusters. So the Avastin, the Humira, and the Rituxan, they are very important pharmaceutical proteins. So to develop the new, the potent antibodies is very important work. So antibodies consist of two heavy chain and two light chain. So generally, this part, so variable, this part is the variable region. So variable heavy chain and variable light chain, they are responsible for the antigen binding. Okay. The other part, so they are just the constant domain. So they are responsible for the antibody structure. But now this part, the variable heavy chain and variable light chains. So this variable region is responsible for the antigen binding. So to change the antibody specificity, to change the antibody affinity, generally these variable region, they are randomized. So by the randomization of this variable region, and then by the screening, we can develop the new antibody which can bind to the new target, and which has a high affinity to the new target. So like this. So from we can construct the antibody library. So the antibody library, and then this is bacterial cell, so tetracell or tetrarchate, the big size of the antibody library, and then they are cultivated, and then add this dye, so antigen, the first dye conjugate to the antigen, and then they mix with this library, and on the library, if the cell they produce some specific antibody, they can capture the antigen, so and they can give a high fluorescence. If the cells they produce a non-specific antibody, they cannot capture this fluorescence dye antigen, so they cannot give high fluorescence. So they can be distinguished. And then also after labeling with this fluorescence dye, and then they can be screened by the flow cytometer. So only the high fluorescent cells, they can be screened by the flow cytometer with very high speed. And then just one time screening is not enough. They are repeated. So one time, and second times, and third times. By the repeating of these cycles, and then actually we can isolate the potential antibodies from the library. So by the repeating this step, so cultivation, the labeling with the process probe, and then screening by flow cytometer, and then cultivation, the labeling, screening, cultivation, labeling, screening. So by repeating this procedure, so the positive clone, which have a high affinity to the target molecule, the population can be enriched. So this is the original library. Their process signal is not high. But after first round, so already increased, the signal or population is already increased. And after second round and the third round, more and more the high fluorescent cells, they were enriched. They are supplemented by the flow cytometer system. So from the library, the high fluorescent population can be enriched more and more. So and then finally, we can isolate the best clones, which has a high affinity to the target antigen. So this strategy was applied to the isolation of antibody against these two viral targets. One is H1N1 influenza virus, and the other is FMDV, the foot and mouth disease, foot and mouth disease virus. So they are very important issue in Korea now. So for the first swine flu, so the H1N1 influenza virus, so we use the N1 epitope. We don't use the whole virus, so just we use the N1 epitope of the cell surface. And then the N1 epitope, they are able to conjugate the fluorescent dye, and then from the antibody library, so the high affinity antibody, they were screened. So so by repeating this screening, so like this, they are repeated. So cultivation, labeling, and screening one round, and the second round, third round, and then the high fluorescent population, they are enriched more and more. So for this swine flu, So first round, they shows very low signal, the red color. Next is the second round is orange color, so a little increase. And then green color, so more high population. And then blue color, so much more high population. And purple color, most of them, they have a high blood signal. So by the 5 times repeatings, so we could isolate the antigen-specific antibodies. So this target, the H1N1, the specific antibody could be isolated by this screening. So finally, the isolated antibody has high affinity, about 11 nanomolar affinity to the target antigen, the H1N1, the virus. The next model is the FMDV, the food and mouth disease virus. And also we performed the library screening. The first round, so red color, very low signal. And then the orange color, so high signal population. And green color is the most popular, highest population. So just by 3 times repeating, we could isolate the best antibody for this target, the FMDV, the virus. Also the isolated antibody has a higher affinity for nanomolar. So like this, so from the library, the antibody library, huge library, we can isolate some antigen-specific antibodies. From the random library, so we could isolate the best antibody for the new target. And also another example of this strategy is affinity maturation of antibody. So we succeed in the isolation of a new antibody. So this antibody can bind the new target. But sometimes we need to improve the binding affinity. So first isolation, the antibodies, they have some good affinity to the target antigen. But sometimes we need to improve this affinity. That's the affinity maturation. So enhancement of affinity. Also, we can use the same strategy. So variable regions are randomized, and then we can isolate the new antibody which has much higher affinity to the target antigen. So in this example, the target antigen is the anthrax. Maybe you know where. So anthrax is a very serious disease which has a high mortality rate of about 80%. So Bacillus anthracis is Bacillus bacteria. So Bacillus anthracis bacteria, they produce some endospore, and this endospore is similar like some white powder. The anthrax toxin, so if they're toxic to the human and animal. So if you inhale the anthrax toxin powder, you will die just in 4 days. Really toxic. So this mortality rate is about 80%. Most of them will die in 4 days. Okay. So another important serious problem of these anthrax they can be used for the biological weapon. Okay. So it's effective as a biological weapon because of its easy of storage and its long lifespan as a spore. So they can be used for the biological weapon. So actually, maybe some of you know this situation. Maybe you know what's this? 2001, so there's the terrible terror in the United States, New York. So the pride, the strike that is the Twin Building. Okay, the Twin Buildings are destroyed. After that, there is another terror. You know, after that, so this envelope was delivered to some people. This envelope contains anthrax toxin. So when you receive this the envelope, maybe you open this envelope, and then the anthrax toxin they spread out, and then you inhale this anthrax toxin, then you will die. So actually, the 5% will die. 5% that die by this terror. So they are very the toxic material. So to protect this toxicity of the anthrax toxin, we need to develop the antibody. So let's see the mechanism, the toxic mechanism of this anthrax toxin. So anthrax toxin produce, and bacillus anthracis, they produce three toxin. So PA toxin, and LF toxin, and EF toxin. And then amongst these three toxins, the PA toxin, first the PA toxin can interact with this human cell. So this is a human cell, and this is a receptor of the human cell. So first the PA toxin interact with this receptor on the human cell, and then they make this some the channel, so the channels, and then through this channel, two other factors, LF and EF factor, they can enter to the host cell, the human cell, through this channel, and then they destroy the host cell, the human cell. Okay? So to protect this anthrax toxins, so we can block this that interaction. So PA toxin interact with this the receptor, but if we block this interaction between the PA toxin and then the receptor, and then the PA toxin cannot make this channel, so the two factor, LF factor and EF factor cannot enter to the host cell, so they don't have any toxicity. Okay? So how to block this interaction? We can use the antibody. So we can develop the antibody which can bind to this PA toxin. So this antibody can bind to the PA toxin. So by the binding of this antibody to the PA toxin, now PA toxin cannot interact with the receptor. So they cannot make a channel, and then the LF cannot enter the host cell. So previously, so this the antibody has been developed. So this 14B7 has been developed for the binding to this PA toxin. However, this affinity is not high, 4.3 nanomolar. It's not bad, but also it's not high. So we start to engineer this 14B7 the antibody to improve the binding affinity to the PA toxin. So same work, the antibody was randomized. So variable results, so they were randomized. So we constructed this random library, and then we added this the probe, so body P, the fluorescent dye, body P conjugated with PA toxin. So and then they mix with this library. So among the library, so if a cell produces a high affinity antibody, so they can interact with this the PA toxin, the body P conjugated with PA toxin, and then they can give a high percent signal, so they can be screened by this flow cytometer fax system, and then they are repeated. So after several rounds, so we can isolate the potential antibody which has a high affinity to the anthrax toxin. So using this strategy, we did a screening from the library, so it has a very low signal. And after the first round and the second round, so we could get this high percent population. And then after the second round, they spread on the agar plate, and then by the individual analysis, we could isolate the two mutants, so M6 and M18. So that this is wild type. So M6 and M18, they show a much higher affinity to the compared to the wild type antibody. So by the affinity analysis by Biochemistry, we found that these isolated mutants, they have about 120-fold the high affinity compared to the original antibody. So compared to the original antibody, these two mutants, they have a much higher, 120-fold the high affinity to the target, the antigen, the PA toxin. Also, from the sequence analysis, the mutation point, so by the mutation point were analyzed, and then the further analysis, so we found that these two mutants, this mutant is very critical for the high affinity. And then the potential of this antibody, isolated antibody, were demonstrated, were validated by the animal test. So we used the rabbit. So to the rabbit, so first we inject the anthrax toxin without antibody. Just the anthrax toxin was injected to the rabbit, and then as you can see, so just PBS without antibody. So when the anthrax toxin injected to the rabbit, so all of them, they were dead in 4 days. So really. And then, well, if we add the antibody, so 1.25 milligram or 2.25 milligram or 5 milligram, so as we increase the antibody, the injections, the survival rate is also increased. So in this case, triangle. So when we use the 2.5 milligram of antibody, so about half of them they can survive. Okay? When you use the 10 milligram of the antibody with endotoxin, so about 90% the rabbit they can survive. So this means the antibodies, the isolated antibody has the high potency to this endotoxin. So now this company uses now they are performing the clinical test of this antibody. Now the clinical test, phase 2, the clinical test, maybe they will commercialize soon. So, okay, that's the first strategy. So using the direct interaction between the protein and the target molecules, we can isolate the protein mutant. The second step is the direct labeling of product with the process dye. So in the first strategy, the target protein, they directly interact with the target molecule. For this case, this is the enzyme. So the enzyme can catalyze this reaction. The substrate is converted to the product. So now we would like to engineer this enzyme for the much higher activity. So we would like to engineer this enzyme. So here's the product. So in this strategy, we add also the fluorescent dye, and then this fluorescent dye can interact with the product, not target enzyme. So this dye can interact with the product, and then they can give some fluorescent signal. So if the enzyme has some high activity, the enzyme has a high activity means the enzyme can produce more product. Okay? So the high activity enzyme, they can produce more product. So the more product means they can give more the higher fluorescence. So fluorescence intensity of the cells are highly correlated with the concentration of a product in the cell. So high fluorescence intensity means the higher activity of the enzyme. And then the low fluorescence intensity means lower activity of the enzymes. Okay? So according to the different enzyme activity, they can give different fluorescence. So by the screening, we can isolate high fluorescent chrome. So high fluorescent chrome is they have a high activity. Okay, so from the library, so we add the fluorescent dye, and then they have a different fluorescence, and then use the flow cytometer, so we can isolate the best clones, which has a much high activity. So in this example, this strategy was applied to this engineering. So engineer will PHB, the polyhydroxybutyrate, the synthesis enzyme in E. coli. The PHB is the biodegradable polymer. So polyhydroxybutyrate is a kind of polymer. So this is the unit of this PHB. They are polymerized. So they are produced like this in the bacterial cell. So they can make the big granule, white granule. So they are some polymer structure. So after cultivation, so this granule can be extracted, and then using this polymer, so we can synthesize this plastic, the bioplastic. This is not. This is a different commercial plastic. So they are developed by this biopolymer. So they can be degraded in nature. Okay, so when the bottle, because that's a simple bottle, the water bottle, so you take the water and then just throw away. And then this bottle can be degraded in nature. Okay, it's very environmental, very important material. So the wild-type E. coli cannot synthesize this polymer, for PHB polymer. to synthesize the pH polymer in the E. coli cell, so we should introduce this enzyme, the genes from the last PAC, PA, PAB genes. They are introduced to the E. coli cell, and E. coli cells, these three enzymes, they can synthesize this polyhydrogen from the acetyl by using these three enzymes, this polymer can be synthesized. So in this synthesis, the product is a PHB. The product is a polymer, the PHB polymer. So to produce more the PHB, the molecules, we need to engineer the synthesis gene, synthesis enzymes, the PAC, PA, the B. So to engineer these enzymes, so we need to label this product, the PHB. So we need to find some process dye which can interact with this biopolymer. So we spend two dyes, body P or Nile leather. Body P is green color dye, and Nile leather is red color dye. They can interact with this PHB granule. And then after some the experiment, we found that the body P can give much higher fluorescent signal than the Nile leather. So we chose the body P for the labeling of this PHB. So these three genes, they are introduced, they are randomized PAC, as one critical enzyme. So this PAC is randomly mutated, and then they are introduced this E. coli cell, and then so they are randomized, and then this big side library is constructed, and then they are cultivated, and then we added this body P dye. So body P dye can interact with the polyhydrogen PHB granule. So from the library, so if the clone has some high active enzyme, they can produce more PHB. They can produce more PHB, and then they can interact with the PHB product can interact with the fluorescent dye body P. So they can give much higher fluorescent signal. Okay?
And so it will be 5 minutes.
So they can be screened by flow cytometer, and then so by repeating the screening, and then we can isolate the potential enzymes. So by the screening from the library, so one time repeat the second, third, fourth, fifth round, so the high population, highest population, they are enriched more and more, and then using this screening, also we could isolate the very strong enzyme, very strong enzyme. By using this enzyme, the PHB, the content is incredibly increased, about the tenfold increased. So the next, the last step is the back screening with the genetic circuit. So this case, the enzyme, so enzyme catalyzes this reaction, so product is produced, and then so here's now this product can be interacted with this transcription factor, and then this transcription factor can be activated. And then activate the transcription factor can also activate this promoter, and then by the activation of this promoter, now GFP gene can be expressed, and then GFP gene, GFP, they can give high fluorescence. So this resembles the electric circuit. Maybe you know this electric circuit. All electric lines, they are connected where this bulb can give light. So like this. So like this, all steps they are connected well. So enzyme catalyze this reaction, and then the transcription factor binds to this product, and then the promoter is activated, and then GFP gene is expressed. They can give a high fluorescence. So all systems, when the all systems works well, all connected well, and then they can give the high fluorescence like this. So using if the enzyme is strong, the enzyme is highly active, they can produce more product, and then they can give more this transcription factor activated, so more GFP gene can be expressed, so they can give much more the green fluorescence, high green fluorescence signals. So using this genetic circuit, so we can isolate more strong the enzyme, high active enzymes. Okay. So this is one example. This enzyme catalyze this reaction, and then by this reaction they produce a superoxide, and then this SOX-R can recognize this superoxide, and then they activate the SOX-S promoter, and then GFP can be expressed. So if the enzyme is strong, they can produce more superoxide, and then more SOX-S promoter can be activated, and then more GFP can be produced. They can give much higher pulse signals. So using this genetic circuit, so we can isolate the high active enzymes. So similar strategy. So like this, using the genetic circuit, and also we can do the protein engineering. We can isolate the more active enzymes. So like this, okay, let me summarize my talk. So using the protein engineering, so we can change the protein properties, protein function. Okay? So there are two approaches, random design and rational design. So the both are okay. So if you have the good structure information, you can use the rational design. But if you don't have any structure information, so you can try this random design. Also sometimes they can be combined. So rational design and random design. Okay? For random design, so you should design the useful screening systems. Okay? Today I showed several screening systems. Also you can use some different screening systems, using those random design screening systems. So all these properties can be changed. Okay? So maybe you remember this slide. So to find the new enzyme, so should you go to the C? We don't need to go to C. Okay? So please come to the lab. You can create this enzyme, new function enzyme. You can don't go to the C. Please come to the lab. Now you can create this. You can save the time. Okay? So this is the take-home message to you. Okay, that's my student. So for my student, about 16 students and one postdoc. So they give a very great contribution to this work. And also Professor George Jordan, the Pion's team. So also we collaborate about antibody engineering. Also I thanks to this financial support. Okay, thanks for your kind attention. Thank you very much. Terima kasih.
Terima kasih. Thank you very much. Okay, Professor Kijun, you may be seated in the you can stand up or you may stand. Okay, so I think now is the time for the Q&A session, but I want to ask Professor Kijun first. You mentioned about the rational design and the random design. You have enough information you can do this rational design for protein, but if you don't have this information, of course you can do random design. But which one is more cost effective provided you have this enough information to do the work?
Okay, so very difficult problem. so I'm not the two approaches, we can use the two approaches. Even though we have, as I mentioned, even though we have some structural information, sometimes that's not correct. And also, even though we have some good structural information, it's not easy to predict the critical point. So that even though we have some structural information, many times we should use random approach, random design approaches. But sometimes, random approach is also some. Today, I showed some successful results, but also I have already some failed. Also, I failed many times because also in this case, we should use some expensive instrument. So flow cytometer, I use the flow cytometer, but this is very expensive. So many scientists cannot use this flow cytometer. But so without this instrument, so they should also need some long time, also several more. Try and error system. So it has some advantages and also disadvantages. But generally, I'm not a rational guy, so I like the random design.
You like the so this for the Q&A session. So for the first time, I would like to ask the three people who want to ask. So first, please talk again over there. Please mention your name and your host institution. Maybe. The mic, you can put it on.
I will introduce myself with Korean methods to Mr. Kim. 안녕하세요, Mr. Kim. 제 이름은 마지기예요. 예, 프로페서 정. 안녕하세요. 제 이름은 마지기예요. 저는 모동이라고 불러요. 프로페서 정, 약올림에서 나서 먼저
You can ask your question.
Okay, I have two questions. First, what do you think about MERS virus in Korea? How to we avoid and cut the
Stop the infection, you mean, maybe?
yeah, not only infection, but stop the infection to go anywhere.
Of the pandemic or the
Yeah, the pandemic.
So it's only isolated in South Korea.
Okay, and two, do you use software to help your protein research? Software in computer?
Sorry, so I can understand it. So,
Do you use software in computer to help your research?
I think he's asking something about whether you are conducting computational approach to help your experiments. I don't know.
computational approach to
Yeah. For your experiments. Computer modeling or did you use any?
So the modeling approaches, so the kind of the rational design.
Yeah.
So first I never responded the random design. So there are many some modeling tools. So based on the protein structure information, so we can predict some critical positions. So the protein model and the target model, so they are docking systems, so the modeling system, modeling program, so they can be docked in the modeling program. So here's the protein structure, and then substrate. So in the modeling program, so we can change some individual residue, and then they can calculate their binding affinity and some thermodynamicity. So all are calculated by some computer program, and then they can find which amino acid is best for the binding to the target molecule, or which amino acid is critical for protein stability, or which one is critical for the protein structure confirmation. So they can be predicted by that.
Thank you. And first question. What about virus?
There's a virus. So if we have some antigen molecule for the MERS virus, we can use this strategy. We have the antibody library. So if we have some epitope of the MERS virus, so we can use the same strategy to isolate the new antibody which can bind to this target molecules. But I think in this case, first the protein is exporting the vaccine, so the first we need to develop some vaccine. So this is the prevention. But next is we need to develop the antibody molecules. But also antibody development takes some long time. Today I showed several examples of antibody engineering, but they are just a candidate, not a real drug. So even though we develop new antibodies, they cannot apply to the real environment.
Okay, I have one
I think this is your last question, please. We are running out of time.
Yeah, what do you think about antibody to fix HIV, fix suspect with HIV virus?
HIV virus.
HIV virus.
Yeah.
So
he's asking whether
you can if someone suspect HIV virus, can you use your method to help?
So for HIV virus, so can you develop the new antibody for the HIV virus, right? Yeah, also same story. So if so also we tried this. From the HIV virus, we can isolate. If we can isolate some epitope, so with the viral antigen, so maybe we can use this the epitope for the antibody scanning. But I'm not sure this isolated antibodies are effective on the HIV the treatment or not. So because we can isolate the antibodies, can bind to the target antigen. But just the binding to the target antigen, that it does not mean we can kill the virus. No. So we can just isolate the antigen binding antibody. But this is just kind of a candidate. So actually they cannot be effective on the virus treatment. So there's more study about it. But we can develop some candidate.
Okay.
Anyone want to ask? Well, I think I saw you first. Okay, you may have. You can go to the mic. I think the committee is going to give you the mic. Okay, please, your name and your institution. You are going to ask. I over that session.
Thank you. Let me introduce myself. My name is Daniel. From Bogor University. I just want to ask one question. I have gotten a task to looking about high throughput screening. May you explain the principle about that and maybe all about the high throughput screening because I just have a little information about that. Just high throughput screening. Can you explain the mechanism, the principle about the high throughput screening? High throughput screening.
High throughput screening.
What is the
principles of high throughput screening?
Yeah, so what is the
he wants to know about the principles.
That's
the mechanism of the working of high throughput screening.
The working mechanism.
The working mechanism of high throughput screening.
HTS. He is asking about the mechanism of high throughput screening.
The mechanism of high throughput screening. Right? So today I use the pro-scientific systems. So these pro-scientific systems they have us the radar system and detector systems, and then the radar will give us the wavelengths, and then fluorescent dye they can be activated, and then the detection systems they can detect this high fluorescence. Okay, so all the library cells they move in the capillary line, and then the individual cells their fluorescence can be analyzed. And then some clones they give a high fluorescence, some clones they give us on the low fluorescence. They can be distinguished, they can be analyzed, and they can be distinguished. And then the high fluorescence cells, they can be scanned, they can be sorted, they can be captured by this fluorescent system. The bacterial cell they have a negative charge, The bacterial cell surface is a negative charge. So when the negative clones or
negative clone row plus clones they clone row. This capillary line is a negative charge. Okay, so they so flow away. However, when the high plus cells pass this capillary line, now the capillary charge can be changed to positive charge. You know? So bacterial cell has a negative charge. But when the positive highly plus cells pass this capillary line, the charge of this capillary line is changed to positive charge. So they can be captured by this process systems. So in this capillary line, they can move very fast. So 70,000 cells per second, and then every time they can be detected, they can be analyzed, and then positive clones they can be captured by this relative charge change. So we can do the isophosphate using this process system. That's the mechanism of the process screening.
Thank you, sir.
Okay, thank you. So I'm going to have another question from that section. Okay, so you, okay. Your name and your institution, please.
Okay, hello, I'm Reza. I'm from Biology Department of Universitas Indonesia. So I want to ask you about what do you think the potential of protein engineering that produced from fungi, for example, like yeast or mold? Because you were explaining mostly the protein engineering from bacteria, right? Like E. coli and Bacillus anthracis. So what about fungi? What about the potential of using fungi as protein?
Using
the fungi, for example, like yeast, it consists of yeast.
So today I introduced some for the protein engineering, the case of the random mutation, random design. So for the random design, also is to construct a big size library. Big size. So big size library can be constructed by the bacterial cell because the transformation efficiency is very high, particularly E. coli cells. So among the many different bacterial cells, we prefer the E. coli cell because the transformation efficiency is really high. So we can construct a big size library. If you use a big size library, you have more chance to isolate the target mutant. But if you use the different bacteria, the Bacillus or Pseudomonas, the transformation efficiency is not high, so it's easy to construct a big size library. But yeast, the Saccharomyces cell is the case. So also we have good tool to transformation to the Saccharomyces cell. So we can construct a big size library. So sometimes also we can use the Saccharomyces cell to the yeast library. Okay. Generally, someone use, in my case, I use the bacterial cell library, bacterial host, E. coli host, but someone also use the yeast, the Saccharomyces library. It depends on your target molecules. If your target molecules, generally we use the bacterial cell, so because it's much simple and then the cultivation is much easier. But some protein, they cannot be produced in the bacterial cell. Yeah, so some protein they require glycosylation. So this glycosylation only can be done in the yeast cell, not E. coli cell. So if your target protein requires this post-translation modification, that glycosylation usually use the E. coli cell library. Okay? But if your target protein does not require this glycosylation, for example, so you can use just E. coli cell library. So it depends on your target molecules. So there are good tools for the yeast cell library. So there library also can big size library, can also big size library.
So it is possible to depend on the target molecule.
Yes.
Thank you very much.
Okay, I want to ask the committee how many times.
But we cannot use the mammalian cell or insect cell host. Just two kinds of bacterial coli cell and the yeast Saccharomyces.
Okay, because we still have 10 more minutes, so I think it is possible for 2 or 3 persons to have. So, anyone who want to pose questions? No? Anyone? Okay, you. Your name and your host institution.
My name is Pro. I'm from Physics Department of ITB. So my question is, in protein engineering, can you use this to create some kind of all-in-one food? Like for example, many food our body requires some certain essential proteins, right? So can you like simply engineer all of the proteins that essential proteins that all humans need, and then create some kind of food from them?
So let's say again. Say again.
I'll say it again.
Yeah.
Okay. What I mean is, humans need certain proteins for their daily needs, right? So can you simply create some kind of engineer some kind of proteins, some kind of food that already contain all the proteins that humans need, and do you get what I mean?
She's saying whether it is possible to create some kind of all-in-one protein engineering that can provide the human nutrition, all human nutrition. All enzymes, everything.
Yeah, like for example, some people have deficiencies in proteins and enzymes that they need, right? Can you kind of decide, can this protein engineering principles devise a food that can provide for all those deficiencies?
So there are so many different enzymes. Of course, I don't know about the case, but so there are so many different enzymes, so we should use some different strategies. So this is just some example of the protein engineering. Also, it depends on the target, the enzymes, the characteristic of the target enzyme, the proteins. So we need to develop some specific system for the target the enzymes. Of course, we need to understand some characteristic of the target enzymes, and then based on this characteristic, we should develop some new screening strategy. So today I showed several strategies, but using this strategy, we cannot engineer all enzymes or proteins.
So there's a potential to go to that direction. Okay, thank you.
So I think for one more person, anyone? No, I think we still have some minutes now. Anyone who's going to rest? We already discussed about a lot of things about rational and random design of proteins, concerning disease, and about all-in-one protein design. Anyone who is going to ask before I give my concluding remarks, please. Okay, please, you. Name your first institution and of course your name.
Thank you for the opportunities. My name is Muki, and I represent the chemistry department. What I would like to ask is, some people is intolerant with lactose. Lactose, yes, is correct. Like we can drink milk. Some people can do that. Some people can. So as far as I know, there is some kind of intolerance in lactose. This kind of engineering, can this protein engineering develop some drugs to cure the intolerance?
So I know what the lactose problem. So some the Asian people doesn't have it's kind of lactate. So lactose cannot be integrated. So when they drink some milk, they cause some diarrhea.
Diarrhea or some
You mean that by the protein engineering, so very difficult to work. So the problem is that some Asian people, they don't have this enzyme. Okay? So one way we can introduce this enzyme to the human, so by some DNA delivery systems, maybe we can deliver some enzyme DNA for these enzymes. Maybe they can produce the lactase, and they can produce the lactose. So we have the enzyme, the lactose. Just to use, in that case, just to use this enzyme. So we don't need to engineer this enzyme because its lactate is very powerful enzyme. But so the problem is they don't have this enzyme. So that's a problem. So they don't have. So we can use this enzyme for patient. But in that case, I think we don't need to modify this enzyme.
I think that this information, this kind of technology can develop something like synthetic protein to help the person with intolerant lactose. Because as you know, milk is something that we need to customize to increase the health of something. So it's like for babies, milk is very important to them. Some babies lately, most of them is practically intolerant in lactose. It's a very sadly actually. So it needs some kind of technology, some kind of protein engineering to develop something like a protein synthetic to help them. Maybe maybe got a pill or something like. It's not some kind of drug, but something that baby can consume that is not to affect the health of the baby.
Maybe it's just some remarks, something to solve the lactose intolerant problem by using protein engineering. It is possible. Making it concise and to the point. The question is all about the protein engineering and solve the lactose intolerant problem.
I'll try to make it simple. So, is there any research? Actually, I don't know. That's why I asked you. Is there outside there any information or research already developed to cure or to help the person intolerant lactose? Not to correct. Maybe the right word is to help them. Is there any research? Yeah. Is there any outside there? Is there any research developed?
That's not some treatment assistance, but some of the swimming because swimming does not contain lactose.
The committee is giving us a chance for another question. One more person is possible. Anyone want to ask? Okay. State your name and your institution.
Yeah, thank you for the chance. My name is Gazi from the Chemistry Department of Universitas Indonesia. I want to ask you, Professor Yun, what's the difference of antibody, general antibody, and monoclonal antibody? What is
Monoclonal antibody and
the general antibody?
General antibody. What's the general antibody?
I have this is antibody and just antibody and monoclonal antibody. What is the difference between
You mean is polyclonal antibody?
Polyclonal antibody. Polyclonal is some mixture of the antibodies. So when you inject some animals, they produce some immune response, and then from the blood we can get some mixture of the antibodies. That's the polyclonal antibody. So polyclonal antibody contains some specific antigen-specific antibody. But monoclonal antibody is just a single antibody, not a mixture of antibodies. So from the polyclonal antibody, we can isolate some very specific antigen-specific antibody. That's the monoclonal antibody. They all have the same shape, but the polyclonal antibody is the general antibody is a mixture of the antibody. So among them, it contains some specific antibody. All it contains a non-specific antibody. This is polyclonal antibody. But, so the more specific, the monoclonal antibody is just a single molecule. which is the higher target molecule, target antibody.
And then by using your daily engineering, how many antibody you can create? My question is, most of your producing is monoclonal antibody or polyclonal antibody?
So in my case, we use a single molecule of antibody molecule. So this is kind of a monoclonal antibody. Just a single molecule, they are mutated, and then we also isolate the middle of the best one. So this is also a monoclonal antibody. But in the general, so we use just antibody. So in my work, for screening, we don't use the whole antibody. Just we use the barrier design. The barrier design is responsible for the antigen binding. Just a barrier free chain and barrier light chain. Just we use this barrier reader for the screening. We not monoclonal antibody, not fluorescent antibody. Just we use a small antibody framework. And then after isolation of this antibody, the framework they are the fusion to the fluorescent antibody. So this is also the monoclonal antibody, which is a single molecule, which is a higher target molecule.
Thank you.
Thank you.
Okay, so thank you very much, Professor Young. I would like to talk a bit about the talk and giving by Professor Young. So there are two methods of protein design, the rational design and random design. Professor Young is using the random design and then using a sophisticated instrument process to screen the result in order to produce good quality protein. So all of you thank Professor Young. Kamsahamnida. And to say thank you for the audience for attending this lecture. So, thank you very much.
After this, we will have the souvenir giving session. So we would like for Mr. Kijunjong to come to the front, and we may invite Siti Astari Rahmani
to come
give
on
the
and give the souvenir. And may we remind all participants to stay until the last because we still have another performance.
So don't go anywhere because this will be the closing performance. The first will be given to Mr. Kijunjong as the speakers.
And the second is
For our narrator, Dr. Ari Aditya.
And after this,
We will have another performance. Right now there will be a solo vocal, but now we have a video to show you.
please
Welcome, Metabolism Class 2050.
Besok ya. Hari ini besok Pak.
Ya, Bapak mana? Oh, belum Kak. Besok ya? Oh iya, besok Kak. Oke.
Kenapa?
Masih punya laporan fisik gak?
Duh, udah nggak tahu di mana. Udah ya, gua mau ngurus proposal penelitian dulu.
Coy, makan yuk. Belum makan. Kamu dulu. Ada. Makan itu. Ayo. Ayo, beli. Jadi makan dong.
Ya, ada Eko sih.
Oh, mereka juga sibuk latihan kayak parabol ya? Oh iya, jadi gue sana.
Bersama
Kimiawi 2012 titanium. Aku tak menyesali hari kemarin, aku tak takut menghadapi hari ini, dan aku tak khawatirkan hari esok. Karena aku yakin dan percaya kebersamaan ini tidak akhir. Waktu terus berjalan hingga tak kusadari. Kini aku mulai takut menghadapi hari ini dan mulai mengkhawatirkan hari esok. Aku merindukan kebersamaan kita. Aku merindukan titanium kimiawi 2012. Rindu hadir di antara kita ketika kita masih saling bertemu. Hadir di antara kita yang masih saling menyapa. Namun terkadang tersenyum pun tak sempat, terkadang melambaikan tangan pun terasa berat. Aku ingin kita yang dahulu saling berbagi cerita dan bercanda tawa. Aku ingin yang kita yang dahulu saling menyapa dan dengan senyum saling melambaikan tangan. Aku akan belajar untuk tidak menyesali hari kemarin. Aku akan belajar untuk tidak takut menghadapi hari ini. Dan aku akan belajar untuk tidak mengkhawatirkan hari esok. Karena pagi selalu membawa harapan dan malam akan selalu meninggalkan kenangan. Titanium Kimiawi 2012. Kenanganku adalah kalian dan harapanku untuk kalian.
Check.
Semuanya yang ada di
Metabolism class 2015. We organized this public lecture together with the class of Chemistry Department, class of 2012. And now we would like our supervisor, Professor Dr. Rosna to give closing speech for this event. Professor, would you please come to the front?
Thank you so much to distinguished guests, especially to my two colleagues from Korea Trans Institute of Science and Technology, for Kim and Kim. We are nothing without you. That's the meaning of the song that we sing before. We are nothing without you. Because you are my colleague, you are our father in this case, in this public class. Saya berbahasa Indonesia saja, excuse me, use Indonesian language. Terima kasih banyak kepada Chemistry 2012, all Chemistry 2012. Lead by Mr. Tio. Lead by Mr. Tio. Tia. Yang diketuai oleh Saudara Tia. This class completely different, even though our class are this one. 40 students only, but totally Chemistry 2012 are about 120. Namun cara mereka untuk menyatu, this kind of thing tidak akan terjadi jika tidak di-lead oleh Tiar dengan baik. Kami tidak. Pada saat sharing dengan Profesor Kim dan Yung di lunch time, we discuss about this whole world, dunia ini, Asia, Eropa, Amerika, Korea, of course, Indonesia, Singapore and Malaysia, Philippines and other countries. Apa yang terjadi? We dream of something. Yang pertama, bahwa memang our national population are about 250 million people. Korea mempunyai penduduk sekitar 40 juta orang. Sekarang dia menyaingi Jepang. Tadi kita diskusi demikian because I graduated from Japan, Tokyo University in Sendai. Then I spent some time in Kansas, Lawrence, and went to Taiwan and other country to learn biotechnology. Nah, yang kami tangkap adalah kapannya Indonesia. Waktu saya kuliah di Jepang in this position, then Korea in this position. I have so many colleagues from Korea studying in Japan. Sekarang Korea Institute Korea Advanced Institute Science and Technology sudah ranking 2 di Asia. Di bawah National University of Singapore nomor 2 dia. Tidak ada yang tidak menyetujui, nomor 2 dia. Profesor kita berdua datang dari universitas yang sangat berkualitas di Asia dan di dunia. Profesor Kim and Profesor Jung is not ordinary people, they are extraordinary people. They have charisma. You have charisma also. My class also they have charisma. Kelas ini tidak untuk mengajar nilai dari awal. I told them tidak untuk mencari nilai mengikuti saya, tapi mencari cinta. Cinta terhadap siapa? Terhadap lawan jenis, terhadap sesama. Pada akhir-akhirnya saya meminta cinta kepada tanah air, yaitu negara Republik Indonesia. Kecil tapi bermanfaat. Apa yang kami lakukan itu, Prof. Kim told me about the situation in Korea, bagaimana competitiveness of the young people in Korea, competitiveness of the young people in Japan. We are nothing. Kalau saya mendiri misalnya di S2 melakukan sesuatu yang sangat militan, they did it also in Korea. Mereka tidak pernah, malahan anaknya Pak Jung hanya 5 hours a day untuk tidur. Selebihnya harus belajar 5 jam saja. Nah, kalau saya pikirkan demikian, panjang ini ceritanya, yang tertangkap oleh saya adalah memang harapan ini adalah di tangan-tangan saudara semuanya. Saya berterima kasih karena Anda, teman-teman semuanya datang dari Universitas Indonesia dan dari luar Universitas Indonesia. This will not happen without you. We love you all. Dan juga jika berkenan, tolong makin giat. Memang kalian anak-anak muda, kalau di sini ada Benny, ada si Ninji, dia senang Ninji. Kalau saya mengajar, should be sedikit militan, tidak bermanja-manja. Mereka tidak bermanja-manja. Anaknya dari awal sudah tidak bermanja-manja, 5 years old. Tidak, no manja-manja. S2 saya nangis di depan saya, orang Batak lah. Nah, dengan demikian, once again, thank you so much, Prof. Kim and Prof. Jung. Kami harapkan semuanya ini bisa terjadi untuk tahun depan, karena sustainability of activity ini sangat kita harus pertahankan, karena ini adalah salah satu parameter bahwa mahasiswa Universitas Indonesia, especially chemistry class yang sudah memiliki AUN terbaik di Universitas Indonesia harus mempertahankan itu kualitasnya dengan baik demi NKRI, bukan demi personal. Dalam konteks saya tidak butuhkan tidak butuhkan profesor, tidak butuhkan sesuatu, tetapi selalu membutuhkan cinta. Jadi tadi yang dibacakan oleh surat cinta tersebut belum terjawab saya untuk saat ini. Mungkin nanti akan suatu saat saya jawab dalam bentuk apa. Thank you so much. Thank you to you all, my dear son and daughter. See you then pada saat-saat yang lain. Berjuang selalu, berjuang untuk pribadi, berjuang untuk keluarga, berjuang untuk lingkungan, dan berjuang pada akhirnya untuk negara dan bangsa. It's not easy. Thank you so much. Thank you. See you then.
we close this event, we want to give thanks for all the participants who came and joined until the last. We also want to thank to our media partner, RTJOI FM,
Suara Mahasiswa UI,
Era FM UNJ,
kabarkampus.com,
seputar kampus.com,
kampusupdate.com,
Radio Kampus ITB,
Sayang Tia UI,
seputar event.
and for all those whom we can mention one by one, we thank you all for coming here and participating to this event. Finally, I, Mentari Bioseti,
And I am Suzan Hakim.
see you next year. Assalamualaikum warahmatullahi wabarakatuh.